Good morning, everybody. Thanks very much for attending this Oregon Lakes Association conference. We have these conferences once a year. We rotate around the state. We tend to feature issues of interest in the area that we're in. Last year, we were in Joseph on Wallowa Lake, and we heard about the changes there to the fish management, particularly to try and bring back the sockeye salmon. So here for these 2 days, and especially for today's program, we have a focus on reservoir management, particularly reservoirs there are many of the reservoirs, of course, in the Willamette Valley. So thanks very much to the speakers for coming to inform us about that area. My name is Theo Dreher. I'm the president of Oregon Lakes Association. As you can see on the slide there, we're passionate about lakes, and there are lots of people who are. If you're one of those and you're not already a member of OLA, please consider joining, and please consider becoming active. At tomorrow's lunchtime, during lunch, we'll have a general meeting or a business meeting, and you'll hear a lot more about Oregon Lakes Association and the things we do. But we support graduate students with scholarships. We've done a lot of work surrounding HABs, so cyanobacterial blooms. There's a poster on the on a stand on the bar table at the back right-hand corner that describes a little bit about cyanobacterial issues in the state. That's been a signature area of focus. We sponsored the cyanobacterial Oregon cyanobacterial blooms stakeholder meeting each year. This year it was in, I think, February. We have it around February and March. We're also active in issues surrounding lakes that need conservation, and particularly in recent years, we've been very active around Lake Abert, the hypersaline lake in Southeast Oregon. And there's another poster on a stand at the back there. Later today, CD3 and Ed Rudberg over here has sponsored a mixer or a social hour. So that will start with a bit of alcohol at 5. We can't serve alcohol on campus before 5. But at that time, then in the afternoon, we'll have some interesting items. You can see them on the program. One is a book signing. It'll be over here by Ron Larson, who's, pardon me, just published a book on the biology and natural history of Lake Abert. This is a work that he's been at for a number of years. It has fabulous photography, and Ron is an expert on Lake Abert. So we'll hear about that later today. There will also be an auction for a boat trip, a salmon fishing trip in the Columbia River estuary, the mouth of the Columbia River, with ex-board member Steve Willey. And so we'll do that around 5 o'clock. Let's see. Let's advance. We can do that here. I mentioned CD3 as a sponsor of the social, but we've got a number of sponsors here today, and we're really thankful for that financial support. And they certainly have services to offer, and we'll feature them during the day. They will introduce themselves briefly. So the program, I've mentioned that briefly. We'll get started in just a sec. Ivan Arismendi will introduce the speakers. This is the focus on reservoir management today. We will have a coffee break there in mid-morning, and then lunch. Everything will be served here. The entire conference is here. Restrooms, there's one restroom just if you go out the entrance here and turn to the right, it's sort of behind this back wall. And then there are further ones if you go a little bit further in that direction, past a little shop, and then at the end, turn left for restrooms. Our talks are mostly 20 minutes, so please stay on schedule. You will get a notification with 5 minutes to go. And if you stop fairly quickly after that time, we'll have time for questions. Yeah, I think we're Zoom is hopefully everybody out there in Zoom land is hearing. Can you send a notification perhaps to Chrissy to I think you were on. How can we figure out if people are connected? Send me an email. Send me an email and we can check that out. Excellent. So I think we're ready to start. I just have one more slide that I found. Oops. Oh dear. We seem to have frozen up. Was that not a... Yeah, so before Ivan comes up to introduce the speakers, I found this slide in my computer database. Well, geez, what's happening? Christina Murphy, Christina Murphy, who will give the first talk, was an OLA scholarship recipient in 2017. And we'll hear talks tomorrow from 2 other scholarship recipients. So excellent to see people who have got a little bit of support from OLA continue in this field. So we'll just get started in a couple of minutes. We'll wait till 22 and then we'll get started with the first talk.
Okay.
Where do you want me to go?
Anything's.
I think buns and get you one, and get you some buns too.
Sure.
So probably about $30. So that's 40 out. Oh, that's pretty. The picture's pretty.
There's Corvallis.
All right, good morning, everyone. My name is Ivan Arismendi. I'm an associate professor at the Department of Fisheries, Wildlife, and Conservation Sciences here at OSU. It's my pleasure to start this session about reservoirs in Oregon. And we thought that having a good breadth of topics that will bring a good discussion about this, especially in light of the new court and decisions about managing lakes in Oregon. And we have people from different backgrounds and different perspectives that will bring that little bit of topics to discuss. And it's great to see people in person in these meetings. I'm glad we have a really good attendance. For those that are online, also welcome to the session. So our first speaker is Dr. Christina Murphy. She is a former OSU grad student and also a former OLA scholarship recipient. Chrissy Murphy was working in lakes for many years now, and she's going to bring a perspective that is really interesting and related to the biology and fish and how we can see that more holistically. Christina is now in the co-op unit of the USGS in the University of Maine, and she will be speaking remotely, and I will be passing the slides. So, Chrissy, welcome.
Thanks. Yeah, we'll see how this works. It looks like the @ symbol is a little funny, so hopefully it works okay. But I'm, as Yvonne mentioned, going to talk today about reservoir ecology kind of broadly, and hopefully convince you all that these are issues of interest, not just the area you are in, but also, if you don't mind advancing the slide, to other areas of the US. So actually, I'm a tiny bit further south than you right now, sitting in what considers itself the state of lakes. And most of those lakes are actually reservoirs. And I'll talk about that in just a minute. The examples I'm going to pull from are, of course, mostly from the Willamette Basin, both because of the meeting theme and because, as Theo and Yvonne noted, the Willamette is where I'm from and where I started working in the basin in 2001 for ODFW. And then I did my undergrad at OSU and came back for a PhD focused on reservoirs. I've been in Maine for 3 years now, almost. And I found this slide actually really interesting because I am not the first person to make this journey. And I guess I'm going to use some examples from Maine because, as I mentioned, reservoirs are an issue of interest, not just to Oregon, right, but across the US and internationally. And I'm not the first person to make the jump. John Mooring actually was from ODFW and came to the co-op unit in 1979. And I stole this slide out of a slide deck and scanned it. So I've been looking for an excuse to use it. So next slide. The reservoirs I'm going to talk about when I make generalizations about reservoirs are actually pretty specific. They're the dams and reservoirs in the Upper Willamette and the Mackenzie. So if you were to drive upstream for an hour and a half to 2 hours, you could reach most of the reservoirs that I'm going to use as kind of generic reservoir examples, because the patterns that we see across these 5 different reservoirs that are indicated by the years of construction are all pretty similar. And I want you to note the years of construction. And then I've circled Fall Creek because, as many of you know, that's a unique kind of case study, or was unique, and maybe becoming less unique currently. So do you want to advance? I feel like we've gone back in time with the advancing slides. So I'm going to, as I mentioned, talk about reservoirs a little bit. I'm going to frame the ecological contributions of reservoirs as challenges and opportunities for the different community members that are part of the composition of a reservoir. So how the habitat attributes translate to differences in phytoplankton, zooplankton, and ultimately fish, including fish of concern like juvenile salmonids. And then how reservoirs themselves are able to influence downstream reaches, not just through fish, but through other materials as well. And then some of what I perceive as major gaps in understanding. So one of those being management seasonality. So hopefully I'll set the stage well for the upcoming material. And then we'll talk a little bit about reservoirs in the context of riverscapes. So if you want to go forward 2 slides, probably. All right, so reservoirs. They hold water, but we made them, right? So the whole point is that reservoirs are not naturally occurring. In the Upper Willamette, this means that they have a much greater proportion of edge habitat because they're functionally flooded valleys. In general, reservoirs are really predisposed to species invasions because you've created this novel habitat that wouldn't have existed in the same form otherwise. They can be manipulated in really profound ways. And that, I think, is the most important thing that distinguishes a reservoir from a natural lake. And even an augmented natural lake that has become a reservoir is now subject to management options that are completely outside of the realm of what you can do if you don't have a structure impounding water, right? And that's the take home that you can make for why, even though we call reservoirs lakes, like Detroit Lake, and we often culturally treat them like lakes, they are not lakes, right? Just for that sole purpose of they are impounded and we have options. So if you want to go forward, I just wanted to present what I think is really interesting about Oregon by contrasting it to reservoir systems in Maine. So you'll notice the date of the publication that I've kind of pasted excerpts from here, and it's prior to the construction of the dams and reservoirs that I'm going to talk about. So there were already over 2,000 dams in Maine. Most of them were constructed on natural lakes as opposed to on rivers, but they are very similar in that they're multi-purpose. So you've got flood control, you've got power. They were originally constructed often for log driving. And what's really interesting, and I think what helps set the stage for some of the management talks that are coming up and their impacts on the taxa that are there and the options that we have, is that the reservoirs in Maine were all private. So all of these dams originally were private. And that's very different from out west where they're public entities. What's interesting is actually there's been a transition here towards public control over some of these in the form of municipalities, because multiple purposes are so important to communities. And that means that there has to be someone to take on the task of maintenance and operations, and those are not inexpensive. And there's a talk coming up later on the benefits and costs of generating power, for example. So that's something that plays into the kind of infrastructural part of reservoirs. And all of that matters. You can go to the next slide, because reservoirs occur in a connected landscape. And so who owns them, how they're managed, why they're managed, all matters to the taxa in the reservoir and the taxa downstream, because they are all linked. And I think this is one of the, I hope, biggest take-home messages, that reservoirs are not these unique, isolated ponds or lakes. They have an outlet and they have some water coming in. And so they are very connected, intimately connected to the areas around them. They also are connected to, the terrestrial habitats. And yet, even as they occur, for example, in the Willamette, in this continuous river system, they represent really profound discontinuity. And you can think about that with the serial discontinuity concept. And next slide. You can think about it for how dams vary and why it matters, which I think is better phrased as how reservoirs are managed and why it matters. And so as we move forward through these talks, thinking about reservoir management and what I'm going to talk about today, which is the impacts on the communities and the ecology of the reservoir itself, you can think about reservoir management as impacting processes that are really fundamental to setting the stage both in the reservoir and downstream. And those are things like sedimentation, chemical transformations, water storage, thermal stratification, and dispersal. So next slide. And next slide. All right, so I mentioned that reservoirs present some unique opportunities, and I use opportunities here from whatever taxa perspective we're discussing. So in this case, warm water fishes, which are not native to the systems in the Upper Willamette. And because reservoirs provide lentic habitat that was not historically available, they have very different community compositions than you see even immediately downstream, which is interesting because the reservoirs are exporting taxa. And I'll show you a slide for that later in the talk. Next slide. They also present really unique opportunities for taxa that would have occurred historically and that continue to occur, but now are very different because of the unique characteristics of reservoir habitat. And so, for example, in the Willamette, 144 millimeters was the largest Chinook salmon smolt that was observed prior to dam construction. And the average was more in the realm of 100 to 130 millimeters. Now, a sub-yearling Chinook salmon smolt that reared in a reservoir can reach over 225 millimeters. And a yearling, next slide, can be even larger. And next slide. There are some the reasons for that, and I'll talk about there are also some potential drawbacks, right? So reservoirs present stratification and forage opportunities that are not the same as what's in the upper river system. They're more akin to very low down, where you have more water. And that's because you get that stagnant water. And I know Evan's going to talk about this, and you'll hear it throughout the 2 days, but you have systems that stratify. And so that warm water at the surface and all of that pelagic productivity, which we'll talk about shortly, contribute to an opportunity for growth. Next slide. That even though these systems are generally oligomesotrophic, is really, really different. But it's a double-edged sword, because as you're growing, you may be increasingly exposed to parasites and actually producing more parasites. So I know Jim is going to talk about some of the implications for that, but both the diel vertical migration that we expect, so we've modeled salmon growth and it makes sense, but it comes with these paired risks. Also the taxa, the zooplankton taxa, which are not pictured here, the cyclopoid copepods carry another parasite that I know Jim is going to talk about. And so that foraging, in the same way that it creates opportunities, presents a substantial issue. So if you want to click it, the fish will go swimming. Whee! Producing parasites and gaining them through its decisions about how to forage in these unique habitats. And so the next slide. I just want to show this really quickly because I want to kind of transition into water level fluctuations. And I know there's a talk specifically about this later in the session, but reservoirs have really extreme water level fluctuations in the Upper Willamette. And I think this is what makes them in particular so much different from some of the natural lakes and unique even within certain contexts of reservoirs. So these water level fluctuations, typically extreme water level fluctuations, are thought of as like 2.5 meters. They can be 30 meters or more under conventional management regimes. So next slide. Also to convince you that, as Theo mentioned, issues of interest to areas we are in, water level fluctuations and reservoirs in general are issues of interest to all of science. And so the publications, that's what this is. I just pulled the metrics from 1985 to 2020, and these are 3 times higher in growth rate than the average for science. So you're all in the right room and you're in the right field. And figuring out these problems will be really important moving forward and advancing the science. So next slide. So I want to mention water level fluctuations both on an annual cycle, as I started out, but also intra-seasonally. So within a season, the reservoirs that I'm going to use as ecological examples fluctuate from 12 to 23 meters. And then over the course of a year, that can be from 30 to 49. Next slide. And that has really visible impacts. So we had a lot of pushback actually for some of our publications on impacts of things like draining because, and I quote, draining must necessarily devastate the littoral community. Well, it turns out water level fluctuations have a really tremendous impact on the littoral community, regardless of kind of how low you go. Because if you're doing it with that magnitude and frequency, you can expect some pretty dramatic impacts. Next slide. So this is just to give you a nice photographic example and cover some of the things that we're going to talk about as we look at the taxa. So the change in habitat volume can result in changes to what I'm going to call flushing, which is the movement, voluntarily or not, of taxa downstream. Competition, because you've shifted the volume of water left in the reservoir, thermal stratification because of the volume change. And so you could end up in a situation where there isn't enough depth to stratify, depending on when the water is low. You can expose sediments, and that leads to things like desiccation, which are really important when you think about chemical transformations and the properties of the sediment itself. And then habitat characteristics like temperature, light, and productivity with things like resuspension. And so if you click the slide, everyone look at the Lookout Point Reservoir, and you might go back one time for me just to show it fill and empty. So this is in August and January respectively, and you can see the amount of water that has left the reservoir, right? So this is just a really important thing to keep in mind, and I hope highlights why we as ecologists deeply appreciate the work of some of the hydrologists and geomorphologists who I know are here for the meeting, because they really set the stage. And no, that's perfect. Yeah, so they shift the habitat availability and the habitat characteristics. In this case, water level fluctuations work pretty dramatically to limit the biomass that we see, especially in the littoral. And they appear to select for tolerant taxa. So water level fluctuations as a baseline appear to act as a really strong selective filter. And I'll cover some of the papers that look at that. And then it modifies behavior. And so when I was here, as Theo pointed out in that slightly embarrassing slide from 2017, we were looking at diel vertical migrations, and they weren't happening. And in fact, there's evidence that diel lateral migrations also are not playing a big role in these systems because they're oligotrophic and because there isn't a lot of structure that's different on the edges than deeper in the water column. It also changes the behavior of spawning fishes. So things like those warm water taxa that would normally be associated with emergent macrophytes and like vegetation, you can imagine that their spawning behavior is different in a system that doesn't have the same structure. And then predation, because again, you've modified some of the habitat attributes that are pretty important in some of those processes. So next slide. So these are a couple of the papers that look at both so Margie put together the densities of the macroinvertebrates of some of the Upper Willamette reservoirs, and they are incredibly low. So those numbers are well over an order of magnitude lower than you would expect to see even in a poorly productive stream. So the associations with water level fluctuations that were occurring at the time appeared to impact the community, but also the communities themselves are, again, as I mentioned, the selective filters really, really limited. We also see a difference in exports, where most of the sediment that are coming out, sediments are pretty inorganic. And that matters for things like stable isotopes, because we tried to collect sediment and run carbon, nitrogen, and sulfur, for example, as we were working on food webs. And it's not possible because there isn't enough material of organic nature in those littoral sediments to produce discernible signatures. So next slide. It also means that pelagic productivity really runs the show. And this is from the paper that we recently published on HABs in these Upper Willamette reservoirs, but it also shows some of the same patterns that I know Kurt's going to talk about going from diatoms to potentially cyanobacteria. These systems typically have their highest biovolumes early in the spring. There are some exceptions. What was really interesting is things weren't necessarily synced, but the taxa are, again, really consistent across these different reservoirs. They're just not doing the same things at the same time, likely because of smaller scale processes. And they're not doing what you would necessarily expect in a eutrophic system or a system that balanced productivity across different aspects of the reservoir. So next slide. The zooplankton community similarly cycle really consistently seasonally and are really limited, right? So if you want to look at the specific taxa, I'd refer you to the 2019 paper. But I wanted to show this kind of process where these reservoirs sort of, they do the same thing over and over every year. And the interesting thing is those water level fluctuations appear to be strong enough in their selection for tolerant taxa that you can drain a reservoir and you don't see discernible differences in algae or zooplankton. Fish would be the one exception, and that likely is transitional in nature because they are not able to hold out as well in really small pockets of water, and they don't have resting stages, right? Like some of these zooplankton and phytoplankton taxa that we see doing really well in these systems. And so life history is playing into whether or not taxa are able to thrive under these conditions. Next slide. And as I mentioned, the taxa that are there aren't moving. So because they're low productivity, because there's not a lot going on the sides, everything sort of sits there as far as the zooplankton community. And that's really interesting because that changes ways that we could think about what's going on with the fish community as well. So next slide. As I mentioned, you know, this water level fluctuations probably have really broad impacts, broader than even I have time to cover today. They also probably influence exposure to other parasites, especially parasites that rely on aggregations of fishes. So one example would be anchorworms, and this is a student that Yvonne and I worked with, and she looked at a different copepod parasite that's much more keen to hook to warm water taxa. And then of course, trophic interactions are influenced by water level fluctuation. So if you want to go forward 2 slides, we'll transition to connectivity. So connectivity impacts communities in the reservoir. So the amount of water coming out, and I expect Jim will probably touch on this, it influences what we see going on in the reservoir with things like parasite dynamics, right? Growth. But also what's being exported from the reservoir, in addition to affecting what remains, affects what happens downstream. And so flow is really structuring. It can export sediment, it can export nutrients, but it also can transition the taxa. So I put a picture of hydra. That's something that we see immediately downstream of many of these dams, in particular when they're flushing water that's at the level where we see the productivity of zooplankton, who are just sitting there and then being, you know, swept downstream into the arms of the waiting community, who are then being subsidized. So I'll talk in like 2 slides on that a little bit more, but temperature is one of the biggest probably feedback mechanisms where that connectivity then influences the communities that are remaining in the reservoir and driving the productivity changes that could be associated with outflow. So I want to show the next slide, which is, I mentioned that transition in the fish community. So this is some of the work that we did with Fall Creek Reservoir, looking at the fish community, and this is data from Greg, who's going to speak later, and others, of a screw trap downstream of the dam. And you can see a transition. It's lagged by about 4 years, but there's a transition from non-native fish taxa to native fish taxa, presumably because the native fish taxa can hold their own when the only thing left is a river, and the non-native taxa cannot. And so then you see that really tight connection between what's in the river, what's being exported downstream. And interestingly, it doesn't translate into persistent communities downstream. So even though those non-native taxa were flushed down, for example, I went out with Brian Bangs during one of the drainings, and we found so many small bluegill downstream. It was really concerning, and they were making it into some of the off-channel habitat, but it was in the middle of winter, and they did not do particularly well if the next summer was any indication because they did not, you know, dominate the fish community downstream, even though they were there in really high numbers immediately upon draining. So next slide. This is just to show that relationship where you can see the red is not the black. That's the dam influence on a community of insects downstream. And so less important than the individual taxa is this idea that the water from coming out of the reservoir is really critical in structuring the downstream community. And then the after, so that darker red, is when selective water withdrawal was implemented. That tends to pull from the level of the reservoir that has a lot of zooplankton production. And so then we saw that shift to hydra and net spinning caddisflies and taxa that could really take advantage of those resource subsidies. So next slide. The same things that happen in the reservoir with water level fluctuations can happen downstream because of the reservoir. So patterns of flow relative to natural water bodies, right? And as I just mentioned, those are related to the composition of exports. And the paper in Water Resources Research is where all of that is published. So let's skip the next slide and just go 2 slides forward for the essence of time. So feedback loops are likely really important. And I made this horrendous figure just to show like how little we know and how great the task is of putting together all of these pieces. And I know there's going to be a great talk by Dalton later in the afternoon on like working together to build these more holistic understanding of these reservoir systems. And part of that is because they're pretty complicated in that they are a lot of different processes happening within the reservoir, and that those processes are related to inflow, to outflow, and they're feeding back through different pathways. So you can go forward 2 slides. So I thought it was interesting when I looked at reservoirs in a riverscape, there are 17 papers ever. I think that when we think about gaps in our understanding and ways to move forward, we could think about trying to put together that horrendous figure into real process-based understanding of how reservoirs are connected, not just to those downstream reaches, but also upstream, and not just upstream through salmonids. So for example, in Lookout Point, when they tagged northern pike minnow, who do really well in the reservoirs, they moved upstream to spawn. So some of the taxa that are not anadromous, not as migratory, are still going to be moving material back and forth. You still have macroinvertebrates, right, that emerge and affect the terrestrial communities to the side and also potentially move upstream. And so I think focusing on some of those directional shifts in energy and in taxa that structure communities will be a really important path forward. Next slide. So you can go forward 1 more. This is really focused on flushing, right? We knew that that the deep drawdowns made a difference. So next slide. We knew that it shifted passage timing. Next slide. But what we don't know is how different those responses are because of season. So most of what we know about those Upper Willamette reservoirs from the research that Yvonne and I and others have conducted has to do with water levels that are low in the fall and the winter, and that then refill in the spring and the summer. And I think there'll be talks later today on how those management regimes might shift. But we don't know, at least I don't know, well, what will happen when draining isn't just in the fall. Next slide. And what will happen when reservoirs perhaps aren't deep enough to stratify with these really distinct patterns of really warm water at the surface, but very cold water at depth. So if you lose that depth, you lose that potentially cold water refugia, and that would shift a lot of the dynamics that we see in growth, in parasites, in community composition. Next slide. And all of those ecological impacts, which I put up a slide from my dissertation here just to say, you know, when we looked at draining, as I mentioned, you know, we didn't see a lot of potentially concerning trade-offs that we had expected. And part of that might be the timing. So looking at when sediments were mobilized, what materials were coming out, and then the impacts to the taxa themselves are probably very dependent on the seasonal aspect of what we're looking at here. So even though all of our research occurred in the summer, it was really looking at processes that had happened earlier in the earlier in the water year. We'll word it that way. So next slide. And then, you know, as I mentioned, I think upstream impacts would be a really good place to look too, because that's a really important habitat for taxa like those juvenile salmon. And of course, dams and reservoirs can occur in series. And I think it would be a prudent idea to get a better handle on how, for example, having Hills Creek upstream of Lookout Point influences what's going on throughout that reservoir corridor. Next slide. So that's what I have. I want to say thank you so much for having me, and I am really disappointed that I'm not in the room with all of you and able to have lunch, but Yvonne has offered to make me a floating head through WhatsApp, so I will be available by video. And I miss all of you tremendously, and I look forward to really excellent presentations on reservoirs and some really excellent discussions. So thanks.
Thank you, Chrissy. So we have plenty of time for questions, and I think you have a microphone at your table. You can just go on. And we also want to be monitoring the chat room from people from Zoom.
And if I went too fast, I can go back and slow down. I actually cut quite a bit of material because I always worry that I will get too bogged down
This
in
one
the finer details. So
want to put the question in the chat for you, Chrissy?
sure.
People
Oh,
can
and I see there's one from Mark. Whether the impact of drawdown on macrophyte communities and reservoirs is linked to the differences seen in fish communities. So a shift between native and non-native fish. So no, and that was part of why I showed that slide on the impacts of water level fluctuations. So draining in these reservoirs, if it occurs on a context where there are 30 to 50 meter water level fluctuations every year. And so there isn't a substantial macrophyte community established, you know, that would be changed by that difference, at least during that time of year. So, you know, there are some mosses and some grasses that will kind of recolonize during the winter, but during the summer itself, when you're talking about a 20 meter water level fluctuation throughout the course of the summer, there's not a lot of macrophytes getting established. And so the warm water fishes that are there are likely having to use kind of a rockier substrate than we would typically think of them using, regardless of whether they're straining.
There's a question in the back.
And then
Yeah, you can go first.
Hi, Christina. Thanks very much for that presentation. It was really great. This is Mark Rosenkrantz, and I'm wondering about, I work on a lake that draws down every 3 years, and it's throughout most of the winter. And I'm wondering what the physical impacts on the resting stages of, say, zooplankton or phytoplankton would be, just from, you know, not necessarily desiccation, but from the physical impacts of rain and rinsing on that community, and how that would affect the population.
that's a great question. And we haven't looked at it specifically, but my guess is that even by doing that every 3 years, that you probably have a pretty tremendous ability to affect what's able to live there during the interim periods. And I say that because there's literature on the different species, for example, of Daphnia, And they have different tolerances to exposure to temperature, right? And different tolerances to, well, if desiccation isn't an issue, to, you know, other disturbance related conditions, you also might see different responses depending that are taxa dependent on sedimentation and things that I expect might also change. But I expect have some geomorphologists and hydrologists in the room who could better speak to the physical processes that might be really different on those different taxa. And then there's another question in the
Yeah, go for it.
chat about macroinvertebrates. And yeah, there is data. So I put up a paper there, Margie. This is the Whitmore paper from 2017, and that's in I want to say Water Resources Research. Let me go back to that slide. River research and applications. So that's real data, and it's for all of those Upper Willamette reservoirs and Blue River, and those are real biomass numbers, so 22 to 42 individuals per meter squared. And we actually, the only thing we haven't published is there's a second year of data that we have. We also have macroinvertebrates downstream of reservoirs. So there's a paper from Cougar Reservoir in 2021 in Water Resources Research, and that's on the unintended consequences that occurred with the temperature control. So thermal restoration then drives pretty substantial shifts in macroinvertebrates downstream. So if your question is focused on downstream macroinvertebrates, we have that data. And then there's also ongoing work by myself and Bill Gurth in collaboration with James White and some of the other folks at the Oregon Water Science Center, looking at the impacts of the different flow regimes. And there again, we're seeing some evidence of dam impacts on taxa composition, potentially, that's associated presumably with exports in that same way, because it looks like it's that same shift towards taxa like hydra. But I think that there's plenty of room to learn a lot more about the macroinvertebrate communities, because we were surprised by how little was there in these reservoirs, but we don't have a lot of context for other reservoirs outside of the Upper Willamette. So that's a great question.
Dalton?
Hey, Chrissy.
Oh, Kurt, phytoplankton.
Wait, Christine, there is one from the room, and then we can go for the online.
Hey, Chrissy. Great talk. This is Dalton Hance. So thanks for the shout out as well. So I was actually kind of struck early in your talk. You talked about reservoirs being this novel thing on the landscape because they're man-made, but also because they're dammed valleys. So just the physics of it are different than a other kind of lake. And I guess I've never thought about that. So I'm just wondering, are there any examples in the Northwest of naturally dammed lakes that we can think of as analogs to maybe study the communities that have had a longer time to develop? The only one I can think of is Lake Chelan, but I'm wondering if there's any in Oregon or so that's the question. Thanks.
Wow, that's a great question and a great idea. Because we think too about the aging, where you probably have like hyperproductivity, and they've seen this in some of the papers, right? And then that initial suite of you've killed all the terrestrial vegetation and turned it into material that's been kind of used or exported, and you're so anyway, that brings up a lot of really good thoughts. I can't think of anything because part of it is unless it was something really durable, natural structures tend to move and break down. You think about like even wood and beaver dams, even much smaller examples. Interesting.
Dalton? Well, our lake, I think.
Okay, yeah. But you can't manipulate it anymore. But is it like a reservoir? I guess the flooded part, yeah, the flooded valleys. I mean, there are certainly lakes, natural lakes that can form that way. But they wouldn't be going up and down by 30 meters.
There's one more question in the chat room, and then we can go to the next speaker.
Okay. So I will answer Kurt's and then I'll type in some answers for the Zoom folks, if I can. So what phytoplankton taxa are most important for the grazers in these reservoirs? Many of the colonial diatoms and green algae are large and might not be that edible. So maybe the small cryptophytes or centric diatoms are most important. You know, I'm not totally sure, but I think there's some really cool literature coming out on grazing and how grazing shifts structure. There's also really interesting work that has occurred for some of the major grazers like Daphnia on kind of the importance of variability in their diet and how actually cyanobacteria can make them more resistant to disease if they have some of it in their like feed. So I don't know the answer, but I know it's really interesting.
Great. Yeah, I'm looking forward to seeing that. Thank you, Christy.
We don't have questions, more time. I would like to keep the time for trying to have everyone on time. Thank you very much, Christina. So our next speaker, Chris Caudill and Matt Kiefer, are going to talk about hydrotrophic level and salmonids in reservoirs.
Good morning, and thank you so much for the invitation to come out and share some of the work that has disappeared. I think it's because Dr. Murphy actually said most of what I need to say, so. Well, the work that I'm going to talk about today is, sure. The work I'm going to talk about today is a synthesis of a bunch of work that we've done over the last probably 15 or 20 years in the Willamette Valley on Chinook salmon and steelhead. And I want to do it through the lens of some emerging ideas and concepts on adaptive capacity. Who's heard of adaptive capacity? Okay, good. So this will all be new.
Yes. How they screen shut down.
Yeah. That's all right. So we know that habitats are changing, right? They're becoming warmer. And so there's increased interest in knowing how biological systems are going to change in response to changing climates. And we know that the traits of biological systems can influence those responses, right? I'll add a little bit. And so we know that biological responses can contribute to the population, what is being thought of as adaptive capacity, or the ability of populations to respond to environmental change. And they can do that a couple of different ways. They can be through genetics, through genetic traits, phenotypic traits, or sort of the ecological interactions as they respond to environmental factors. Right. So for the last about 4 or 5 years, I've become sort of blissfully ignorant of what's happening with policy with salmon down in the Willamette, because I've been working on a project with rainbow trout in Idaho to ask how genomic information can help us better understand adaptive capacity. And so those maps are projected suitable habitat for trout currently and in the future. And if we think about those as suitable habitat, we can imagine that species can either adapt in place, right? They can change their traits, allow them to stay in place with changing environments, or they can move in space. And I'm going to focus on adapt in place because we're often concerned with conserving geographic populations, right? Keeping things or communities in the places that they're at. So the idea of evolutionary rescue has been around for a long time, going all the way back to Darwin. This is basically the idea that standing genetic variation or ultimately mutation can allow populations to track changing environments. Pepper moths are sort of literally the textbook example, right? And so this microevolution can allow populations to persist. This is sometimes called genetic rescue, and in the literature, adaptive potential. Okay. We can also think about doing this in a management context through assisted migration, where we actively manage the genomics of a population. Well, populations can often persist in place by what's called plastic rescue, or the expression of phenotypic plasticity. Daphnia are probably a well-known and actually one of the classic examples of phenotypic plasticity. And this is a genetic by environment interaction where the same genotype expresses a different phenotype in two different environments, right? And this can happen through a bunch of different mechanisms, changed morphology, as in this example, physiology, acclimation is a good example. Life history, I'll give an example of, and can happen through either underlying genomic architecture or through epigenetic effects. One of the interesting ideas is that there can be novel phenotypes that emerge as environments change, and that's where this idea of plastic rescue comes from. And I'll talk about an example, the example that Christina talked about with smolts in the Willamette as being a potential example of this. So when we started this large interdisciplinary program called GEM3 to understand rainbow trout and sagebrush adaptive capacity, we stepped back and tried to wrangle some the existing literature and synthesize it into a conceptual framework. And kind of the way we ended up structuring this, or the patterns we recognized, were that this idea of evolutionary rescue is called adaptive potential, mostly in the evolutionary biology literature. If we think about the ecological literature, there are a bunch of processes that contribute to so-called plastic rescue, and that's often called what we dubbed biological adaptive capacity. And again, that's the traits or attributes of a population that allow a population to persist. We also recognized or realized that this idea of adaptive capacity is being used more broadly. If you read the IPCC reports, they use a broader definition that includes the ability of systems, institutions, humans, and other organisms to adjust to potential damage, take advantages of opportunities, or to respond to consequences, okay? And so the social ecological systems adaptive capacity is a broader term that biological adaptive capacity sits under in sort of a hierarchical structure. So what I'd like to do for the rest of the talk is go through 3 case examples from the Willamette, and then finish up by mapping these back onto this conceptual framework and closing with a few thoughts. I'll talk about those life history pathways in smolts, talk about thermal exposure in adult salmon and how that relates to pre-spawn mortality and how that relates to reservoirs. And I'll briefly think for just a few minutes about how genetic management of anadromous fish, steelhead, plays out, and the role of dams and some opportunities for management there. Well, you're well familiar with the Willamette River Basin. There are two main anadromous populations that occur above Willamette Falls, and both of these are ESA listed. I'll talk about Chinook salmon first and touch on steelhead at the end. A major attribute of this watershed now, of course, is the Willamette Valley Project, is a series of dams that were put in place in large part to keep Portland and other cities from going underwater. It currently blocks about half of the spawning and rearing habitat for salmon and steelhead. and holds about a quarter of the annual precipitation potentially. Sort of baked into the authorizing legislation, my understanding, though it's dim, is that hatchery mitigation was intended to be a major part of the operation of the Willamette Valley project. It has implications that are ongoing. Well, the first character in the play here is Chinook salmon. A couple of important attributes is that they come in the spring, in May and June. They hold in freshwater for weeks to, well, for several months actually, and then spawn in the fall as water temperatures come down. That's probably related to being able to get over the falls historically during high enough flows. There's no fish passage at the dams in these spawning tributaries that I'll be talking about. They're listed as threatened. And again, hatchery mitigation. Hatchery fish are clipped so that there can be legal harvest of hatchery fish. Interestingly, historically, there was life history variation in Chinook salmon, and that variation was associated with the timing of when juveniles moved downstream and which habitats they reared in before they reached the ocean. And there were sort of three main pathways. The first was they would come out of gravel in tributaries, move relatively quickly downstream, do a little bit of rearing in the estuary, and go out as sub-yearlings. Some would stay in the natal trib that first summer and move out in the winter as sub-yearlings are right on their first birthday. And then sort of a classic Chinook yearling life history moving down the following spring. Now, for the past couple of decades, adults returning to collection facilities, usually below the dams, if there's been excess hatchery broodstock, those adults were transported around the dam in what's called outplanting regionally. I'll probably talk mostly about it as translocation, released into spawning tributaries to spawn. And the aim was to get some marine-derived nutrients into the system and provide a forage base for also ESA-listed bull trout in those tributaries. A couple of years into this, there were unmarked adults coming back to those traps, and people sort of realized that there was some natural production and fish were able to get through the dams. And so now these above dam populations are an important part of recovery planning. And I'll talk about how that's playing out. So juvenile Chinook salmon, like Dr. Murphy outlined, can grow really fast in these reservoirs. When we came into the system, there was still a bit of controversy about whether or not where these fish were coming from and how old they were. We did a number of studies in collaboration with the Corps and with ODFW, who originally postulated the presence of these reservoir-reared fish from scale data. We were able to use otolith microchemistry to demonstrate that fish were indeed growing in the reservoirs and were also growing quite rapidly. And so we now sort of posit that there's sort of a Chinook salmon are able to express almost a continuum of different life histories on this landscape. And sort of those historical life histories are still maybe present, especially in the undammed tributaries. But then in the dam system without planting, we have these other pathways that represent these reservoir-reared life history pathways. How an individual expresses that life history is almost certainly in part because of genetics, but the management and structure of the reservoir systems have a strong effect. And there are really three big factors. One is where the adults are outplanted, whether they're above or below the dam. The passage structures and conditions at the dam determine how quickly juveniles can pass down and at what age. And then the reservoir operations also have a really strong influence because as the reservoirs are drawn down, that's often the only time fish can get out under current conditions. Okay. And so unfortunately, the smolt numbers that are produced every year in these tributaries and these life history compositions are they're not really well quantified in the system. So one of the things that we've done with the Corps is to do some modeling to ask the question, well, does this life history variation really matter to recovery? And how do we deal with the fact that there's all this complexity? So we built some models where we track each one of these life histories and have estimates for their relative survival. It's kind of a pain in the neck because the adults come back over 3 years and it gets messy quickly. But really the aim is to estimate the relative performance of each one of these life history pathways in terms of survival and how that contributes to what's called cohort replacement rate, which is really just a single generation estimate of population growth rate. Are these populations replacing themselves? And so we can look at each one of those individual life history pathways, ask which ones we think are performing better or worse, add them all up, and under current conditions, the estimates for most of these locations are that they're not replacing. We actually compared these to some genetic pedigree estimates. In some cases, they overlap, some cases they don't. The utility of these models, I think, really is those absolute estimates are tough because there's a lot of parameter uncertainty. But I think one of the useful parts of these models is really asking about different scenarios. And so in this case, we can ask, how do the overall population productivity differ if you put fish above or below? And if you put fish below under current conditions, they do much better. We built a Shiny app so that folks can run through these different kinds of alternative scenarios. And then we can ask, because dam passage facilities are likely to change the composition of those life history pathways for each cohort, what would that likely do to the population growth rate? And here, this suggests that these populations are more sensitive to improving survival for yearlings than subyearlings. If you change subyearling survival at the dam, it doesn't matter much because they don't do very well downstream. One of the early conclusions of this that we're still exploring in simulation scenarios is that there may be a future trade-off where improved passage at the dams changes the life history composition of the fish that goes downstream. And it's possible that you could move fish down so early and young, the conditions downstream cause high mortality, and you actually end up with fewer adults coming back. So that's one of the things that we're using these models to explore. Okay. So the second example is really thinking about pre-spawn mortality, which is of concern in these populations because the adults do this remarkable thing that salmon are famous for. They go over half an ocean basin, get right back to where they were born, and successfully do that, but then perish before they can spawn. In some years, those 60, 70, and sometimes even 80 or 90% of the adults can die. In the Willamette, compared to other basins in the Columbia River, we've noted that those rates are higher and that it's consistently associated with higher temperatures. And so we've been thinking about ways to mitigate the thermal exposure of adults during migration and during that holding period, just quantifying that and then modeling it. One of the things that we came upon, collaborated with Greg on this, was can we release adults to alternative locations, find the coolest streams and places in the streams, or can we release them to the reservoir and allow the fish to behaviorally thermoregulate by going below the thermocline and reduce that thermal exposure during the summertime? So we did this with telemetred fish. This is an example of the depth and the temperature for a fish as it was placed into Foster Reservoir. It was there for a couple of weeks, stayed below the thermocline, presumably picking its preferred temperature. It actually nosed up into the Middle Fork Santiam, which has really cold water coming downstream from an upstream reservoir, came back into the reservoir, and then warmed up as it went up into the South Santiam to finish its holding before spawning. We found that 3/4 of the fish that were put in the reservoir had a thermal benefit, and that their total degree days, their accumulated degree exposure, declined by about 12 or 15%, about 100 degree days per fish, and they saw lower maximum temperatures. Okay. There's an important caveat, though. We found that about 1 in 8 fish fell back downstream over Foster Dam. And so that has a mortality risk and obviously is not so great for doing what you're trying to do, which is outplant fish. However, that might actually be a good thing if fish are trying to home back downstream because they had been from downstream and placed above the dam incorrectly, right? So, and there is some evidence from genetic pedigrees that at least some of the fish were doing that. Okay, so that example actually brings us to our last example here, thinking about integration and what's sort of informally been called using dams to create wild fish sanctuaries above the dams and allow hatchery mitigation below the dams. Steelhead are the other ESA-listed population. The native subspecies is winter steelhead. The taxonomy is challenging in rainbow trout, but it's often, that's the subspecies. It has some life history traits that make it different. It comes in the wintertime to the summer steelhead, which are actually out of basin sort of mongrel genetic hatchery stocks that were brought in for that hatchery mitigation. The hatchery fish are clipped, and the idea is that we know that there is integration between these two stocks based on some genomic analyses. We don't fully understand the consequences, but in most other salmonid populations, hybrids don't do as well as purebred local stocks. And so folks are still sort of grappling with what to do with fish that arrive at one of these collection facilities that has a fin, that doesn't have a fin clip, meaning that it was probably born in the stream. In an ideal world, all the fish would be perfect at homing, and they would all represent the offspring of fish that had been outplanted, and you would just take them up and put them in the tributary. Unfortunately, some of those fish may have just wandered into the trap, and they actually are naturally breeding from downstream. And so there's a potential to actually mine those populations and cause problems for those downstream populations by outplanting them, especially if survival through the dam is low. They might be unmarked summer steelhead. And so then you'd be introducing the fish that you really don't want to hybridize above the dam. And they may be sort of stealth introgressed fish that were the offspring of winter summer hybrids that bred downstream and then returned as unmarked individuals. Okay. So we know that these hybrids exist in the system. The dams actually present kind of an interesting opportunity to think about the genetic management to reduce hybridization by pulling fish, doing real-time genetic analyses to understand what their ancestry is, and using that as a filter for wild steelhead going above the dam only. And there's starting to be some talk to think about how we could use genomic information to identify fish with genetic traits that will be suited for those upstream locations in the future conditions, and sort of pre-select fish for a particular set of conditions above the dam. Okay. So we come back to this conceptual framework. You know, I think this last example fits in with this idea of adaptive capacity and managing for the genetics of a population to be able to persist by being able to track the environment. I think this example of smolts is a really nice example of the potential for plastic rescue, where that portfolio effect of life history traits allows this population to persist despite the dramatic alteration of their environment. And kind of strangely, these dams have blocked much of the habitat available to these species. That habitat is likely to be what's sometimes called a climate shield, the place that is going to be relatively resistant to climate change effects. And so getting those fish up there is adding adaptive capacity, but that clearly involves the social ecological system, right? So this fits into that broader context. There's also opportunity, again, to manage the thermal habitat by placing fish in the reservoir. And if you let me, I'll give one more example when I'm done of how reservoir management can influence thermal exposure downstream. Okay. So, you know, buzzword bingo, right? Adaptive capacity. You haven't heard about it yet. You may or may not, as this sort of trickles in from both the conservation biology and the policy worlds. Is this a useful framework? Well, really, it's kind of just repackaging old ideas. And these come really from the idea of sort of niches and the fundamental versus the realized niche, and some ideas about resilience and from systems theory. I think it has some advantages because many assessments of climate impacts to date have been static. And the advantage of these adaptive capacity frameworks is they explicitly bring in the idea that there are these feedbacks among these systems and that the responses of these systems are likely to not be equilibrial, right? The idea of resilience presumes that you're going to come back to some previous state, okay, since we're living in a non-equilibrium world. And finally, the broadest concepts integrate human intent and actions. That's actually how we define the difference between biological and social ecological systems in our review, was that we have these thoughts and these ideas and then can cause ecological consequences, which is different than biological systems and how ecological systems, natural systems respond. And I think they're helpful for identifying sort of the surprises and the opportunities, as perverse as some of those may be. With that, I'd really like to thank a whole bunch of people, both on the GEM 3 project shown in the picture, but a huge number of people that worked on this stuff over the last couple of decades, including long-standing collaborations with folks at the Army Corps of Engineers and at Oregon State. There's a whole bunch of the stuff that drew from on this. Do I have time for like one more little bit? Yeah. So one of the other things that we've been doing is temperature management is something that folks are thinking about. And so we took our radiotelemetrid fish that were moving up from Willamette Falls, and we had temperature data for their migration histories. We've simulated those histories, and a couple of things that have come out of this. Fish that come in early in the season make it upstream before it gets too warm. Fish that come in late experience really high temperatures in the river, so those maximum temperatures. And that's a thermal metric that can really drive physiological stress and rapid death, right? A critical upper thermal temperature limit. Fish that come in early and beat the warm temperatures early, though, have to stay in those tributaries longer, and they accumulate more thermal exposure. And so there's a cumulative effect. And I think this is an important example because to figure out what the effects are, you need multiple metrics of thermal exposure. And as we think about how these systems are changing, just focusing on mean temperatures isn't going to be enough. Sort of the timing, maximum, and cumulative thermal impacts are three axes that we really need to think about. And of course, we can use models to then think about how temperature exposure would change with temperature control towers, which this isn't going to cooperate to show those examples. Oh. There it is. And so if you add a temperature tower, you can get those thermal cumulative exposures down compared to current conditions. Thank you so much.
Thank you very much. Thank you very much. We have a coffee break now until 11:10. So we have time to ask questions directly to Chris during the coffee break. Thank you very much. Thank you very much. We're going to continue our session with the next presenter, Dr. Jim Peterson from USGS Univ and Oregon State University, is talking about this interesting system about salmonids and copper-boat parasites. Jim?
Thank you. So this is a really involved project. There's a lot of people here. Yvonne and Chrissy was also part of a larger effort looking at a particular parasite of fishes attaching to fishes in the Willamette Basin. So Chris already covered this. We're out planting fish above the reservoirs. We've been doing it for a while, and what we get is natural production. And the juveniles in these reservoirs, different life histories, they get large. And large is usually assumed to be a good thing, right? You make it to the ocean larger, more than likely you'll have a higher survival probability. But really, our reservoirs ideal rearing environments for these juvenile salmon. So around 2010, 2011, ODF&W, kind of led by Fred Monzik and folks, found that there are some pretty high rates of parasitism by this external parasite called a copepod, a parasitic copepod. They typically attach to the gill area. Some people call them gill lice. You can see kind of right in the picture to the left-hand side, those little white things, that's those parasitic copepods. And the question was, is this really an issue? Is this really going to be a problem for these fish? We didn't know a lot about these parasites. So the life cycle of this parasite is that it has one free swimming form. It's a copepod, a copepodid, and it swims around, and Chrissy and Yvonne studied this, and it attaches to a place on the salmon, the juvenile salmon, the host. And these are specific for Onirhynchus. So rainbow trout, Pacific salmon, it's specific to them. They attach to the host, they dig in, they excavate areas, they attach, and then they grow at the side of the attachment right there. And then after a few weeks, typically about 6 to 8 weeks, females get egg cases, and then they drop their juvenile copepodids right there. So typically the only individuals you can observe with the naked eye are these females. They're the largest ones you can see. You observe them. They're also the tip of the iceberg. So we said, well, is it an issue? What's going on with these things? So what we did was we wanted to figure out, are these good, novel rearing environments still for fish? What's the role these parasites are having in the ecology of these rearing juvenile salmon? And what we do is sort of an integrated approach that includes a field component. We want to look at spatial and temporal distribution of infections. We want to look at seasonality. We want to estimate what's called parasite-associated mortality. And we also combine that with laboratory studies where we did experimental infections of these fish to understand the dynamics of the infection. And we wanted to also look at osmolar regulation, right? These salmon, juvenile salmon, are going to make it to the ocean. Can they handle the transition to salt water? So the field research, we have sort of this historic component. ODF&W did sampling in 3 reservoirs, Detroit, Cougar, and Lookout Point from 2012 through about 2016. We fortified that with contemporary sampling, the sort of historic sampling. They only looked at adult female copepods attached to the gills. So we're just looking at the visible portion. The contemporary sampling that Ethan Gardner did in 2020, he focused on Lookout Point in particular, but then he did necropsies. So he wanted to look at what was going on to the gills themselves and actually trying to enumerate some of the juvenile stages and the adult stages. So we also did some statistical analysis. We looked at what's called prevalence. Prevalence is the proportion or percentage of fish that are infected. And then there's also intensity of infection, and that's the number of copepods per infected fish. So it doesn't count the fish that have 0 copepods on that. And then we did this analysis of something called parasite-associated host mortality. And we used the Wilbur, what they call full likelihood method, which is sort of a cutting edge in the field of wildlife parasite diseases. So what we found was pretty strong evidence of parasite-associated host mortality. On the x-axis is the number of copepodids, adult copepodids, so the infection intensity. On the y-axis is the host survival. And you can see with the increased number of copepods on the gills, there's a decrease in survival. However, it was quite variable from reservoir to reservoir and from year to year in terms of what would be the sort of lethal dose. That dotted line represents sort of that 50%. So there's a wide variation in what sort of infection rate can cause mortality in the fish. So we also looked at some of the dynamics. We have the yearlings. Chris talked about the different life history strategies and the sub-yearlings. The yearlings tended to have higher infection intensities, higher infection prevalence than the sub-yearlings. Really, it's a function of time. We also find that water temperatures affected infection intensity, infection prevalence, as well as reservoir inflows, reservoir elevation, reservoir outflows. So if you were holding water longer time periods in the reservoirs, you increased the chances of a fish getting an infection. So reservoir operations, at least statistically on paper, they look like you might be able to do a little something to perhaps reduce infection rates. And we also found through the histopathology that there were all stages of the infection were present at all times of the year on these infected fish. So we had to look through microscopes to find these microscopic stages. Now we had a undergraduate student, she was gonna do look at food habits on these juvenile salmon that Ethan was using for the contemporary field study. And what Marina found was severe infections by a nematode. And Greg, I think this is probably going to be the grossest picture we're going to look at. And all those little spaghetti stuff, those are nematodes in there. One thing about these nematodes is the host cannot shed the nematodes, right? They can't drop them. They don't get over the nematodes. And what we found, and we look statistically, you see through time that the infection rate goes down. So these fish that are heavily infected are either dying or they're leaving. They're leaving the system. And we also see sort of this decrease in with fork length, the size of the fish, which also could indicate fish are either leaving or they're dying from this parasite. So this was really unexpected for us. So the laboratory studies, we did a couple of things. We did infection experiments. We looked at things like the effect of copepod density, water temperature, on infection rates, intensity, prevalence. We also did necropsies to look at gill damage. We wanted to figure out what's going on with respect to these infections. And then we did what's called saltwater challenges, where you expose fish to salt water. That's the same equivalent concentration of salt as there is in an ocean. And we did it at a couple of different time periods. 2 months after they were exposed to the infectious stage, the copepodids, and 4 months after they were exposed to the infectious stage. And then we looked at, can these fish handle salt water? And for a portion of these studies, we also measured blood sodium. And what that indicates is if a fish is leaky, if it's taking salt into it and it can't osmoregulate. So for the infection prevalence portion of the study, we found that water temperature, the higher the water temperature, the higher infection prevalence. Prevalence is also affected by the density of the infectious stage, little copepodids, these microscopic things. And we were able to match the infection rates that we saw in the field. For infection intensity, same thing goes, warmer temperatures, higher copepodid densities. We have higher infection rates, higher intensities. And for some of these combinations, especially the warm and high copepodid densities, we're able to match the intensities we observed in the field study. The saltwater challenge was actually pretty surprising. We did 3 different saltwater challenges here. And Carl Schreck, co-author, one of his big things is looking at the ability of fish to osmoregulate. And in the very first saltwater challenge we did, we had incredibly high morbidity rates, which was really unexpected. This typically doesn't happen. And if we look at the chances of dying, the odds of dying, basically five times higher for fish that were exposed to the copepodids than were not exposed to the copepodids. The warmer water temperatures that also had very high copepodid densities that were, attaching to the fish and perhaps dropping off. It was pretty much off the charts in terms of the effect on survival or mortality. So we also found that simply exposure to the copepodids resulted in lethal levels of sodium in the blood plasma of these fish. So these fish weren't dying. We had a certain segment of the fish that would die or at least show severe stress where we had to take them out of the treatment and the experiment for them. The ones that made it through, we then looked at the blood sodium for them. And what we found was just being exposed to the cobapodids, again, you're having about a 5 times higher chance of having sodium levels in your blood, even if you survive, that you're not going to make it into the saltwater transition. You're just going to die there, you know, buoy 10, wherever it is in the Columbia, you're not going to make it. But we did find one thing that 4 months post-exposure, that there were there's a significant effect of time. And that means that the sodium levels were lower in these fish that had rested, if you will, 4 months post-exposure. So that might indicate there's some evidence of healing that's going on after these fish experience an infection. So this isn't necessarily gross pictures, but in the upper left-hand corner, you can see those are the initial infections by the juvenile stages right there. And those are right there, cobapodids that attached, and they are now undergoing their first metamorphosis as they attach to the gills. And then if you look at the upper, upper left, right-hand side, you'll see the damage. The excavation occurs while they're attaching to there. We found more damage and more damaged sites, and actually filaments of individual cobapodids or copepods that had gone through different transformations but didn't survive. They either dropped off, kind of unlikely, or they just died before they were able to transition into adults. And what we found was, for every single adult female copepod you see there, they've probably been exposed to tens of copepodids, juvenile stages. And the juvenile stages are the ones that are doing all the damage. So even if you're not seeing damage to the gills, if they've been exposed and you're able to do some histopathology, you would find there's damage to the gills and probably filaments of stages that you didn't observe. However, if you look here at the lower right-hand side, you'll see there was some healing that occurred in some of the fish. So they are experiencing the damage. They have the ability to heal. So that's a good thing. So if you take the two different sources of mortality, that's the direct mortality or morbidity, I should say, from the exposure to salt water that occurred very shortly after being exposed to salt water, and you add on those individuals that had the blood sodium levels, they didn't die during it, but they had them high enough where they're not going to make it. You have very high saltwater morbidity. We're talking here between, 50 and 60% of those individuals that make it to the ocean that are exposed to copepods are not doing well. However, there might be a little bit of healing that went on, but it was a smaller effect. So the question is this going to complicate the transition from freshwater to saltwater of those individuals that reared in the reservoir and were able to make it to the Columbia River estuary? Well, if we think about in the Willamette and these reservoirs, then we think about just that sort of yearling life history. We look at some of the data that was collected by ODF&W that looks at the timing of the outmigrants of these yearling life histories right here. Here it is, the studies that Monzik et al. Did that look at infection prevalence through November. And we start counting months between the last time we saw them in December, we know they still have high prevalence rates, to the time when they have peak outmigration time in mid-April. Right, that 4 months, that's that 4 months that we observed. We still have a significant portion of those fish, even if they healed or partially healed after being exposed in the Willamette reservoirs, that are likely hitting the estuary and not making it because they can't handle the saltwater transition. So let me fix this. There we go. So reservoirs as rearing habit. They grow big fish, but there are some hazards associated with it. Right now, I'm working with a group of folks in the Central Valley, California. They're talking about taking winter run and outplanting them in the McCloud, which is above Shasta Lake. And my suggestion is, you better be careful what you want to do with those fish, because this is really unanticipated, right? We really didn't think this was going to be, parasites. They got them, they can make it. And in fact, we're probably seeing fairly high effects on mortality, either in the reservoir, parasite-associated mortality, or just damage to the gills that can affect their ability to transition to saltwater. So the juvenile copepod stages, they cause most of the damage. And when you're looking at fish in the field, all you're doing is counting the females. So think 10 times those numbers, they've already attached to the gills and probably just dropped off at some point, didn't survive. If juveniles do survive the reservoir, there is a reduced ability to withstand salt water as well. So Chris talked about sort of these populations are kind of at or below replacement. And it might be that sometimes these reservoirs, at least in some years when infection rates are high, might act as sinks, right? So you're producing individuals upstream, they're in the reservoir, but there's not enough of them surviving to replace the adults that created them. But there is possible healing, that's right. Acknowledgements, a lot of folks worked on this effort, and funding was provided by the Army Corps, which we really appreciate. And ODF&W, great cooperators on this as well. So any questions? Yeah, you got it, I think.
Here is okay. The mic's on. Great talk. I assume there's a density dependent effect of the population of fish that's causing the infection rate. And have you looked at what that kind of threshold might be for density of fish to accelerate the rate of infection?
We looked at that in the laboratory, but we really couldn't pick up. So we had fish more crowded in a smaller tank and a bigger tank, but we couldn't pick up a tank size crowding effect in there, at least in the lab. In the wild, Chrissy and Yvonne actually looked at the free swimming stages and where they're distributed in there. And I think, if I remember correctly, and if Chrissy's on, you can correct me if you want, that you saw increases in the densities of those copepodids, basically in the zone we were catching most of our fish, like that 16-degree, 15-degree zone right there. So I think they might be crowded in the zone as well as the copepodids, the infection stage. So there might be something there.
One follow-up question: is there interactions with, say, kokanee or resident salmonids that could be affecting the infection rates?
Well, they found some infected kokanee, but not a lot. ODF&W did with some of their work, but they didn't collect a lot of kokanee. So I really wouldn't hazard a guess on that.
Temperature and prevalence. But did you check the runs? Did you check the runs that have no reservoirs? So is the prevalence, or can you find these infections in river run salmon?
Yeah, the original study that was done by Monzik et al. Compared the stream rearing individuals to and it was not even, it's significantly different, way lower. So we're talking, you might find one or two individuals. We're talking prevalence of less than 50% and an intensity of typically less than 3 in the what do you call it? Stream rearing individuals. However, one thing, the adults they outplant, when you look at them, they can handle heavy loads. Their gills are basically furry. When they go to outplant them, Chris can probably tell you this too. I mean, they have a lot of copepods on them. So, the possibility exists that, are we importing, what do you call it, subsidizing the copepodid population by doing that? More questions? Oh. No, I think we're good. Any questions online? All right, thank you.
No, I have a question.
Go for it.
Yeah. I'm curious about the saltwater experiments. That's really fascinating to look at their health and physiology. So I wonder about the effects of being exposed to high pH habitats, and because with a lot of these cascade systems see pretty high pH at times from periphyte and other algal bloom type of situations, and just wonder how if the pH might exacerbate this issue with the copepodids. And that is a hard word to say.
Yeah, it could possibly. That might be an interesting way or an interesting avenue to research, I think, in the future.
Sounds good. Thanks.
All right, thank you very much. So our next speaker is Gwen and Bruce. They're going to talk about western pond turtles, bringing another layer to this really complex problem of reservoir management in Oregon and the Willamette River.
Thank you very much, Yvonne. So thank you everyone who's sticking it out. One more talk until lunch. So hopefully everyone's still at a point where they can pay attention, and I'm lucky because I get to talk about something that's very charismatic. So at least you get to look at pretty pictures of turtles. And I'm going to start off by thanking some of our collaborators. This is going to sound real familiar. This is a little repetitive, but since we're talking about reservoirs and we're all people who are working in reservoirs, probably not surprise you that I'm going to thank the Army Corps of Engineers, OSU, especially Yvonne Arismendi's lab, USGS, Forest Service, right? Kind of all the people we've already been hearing about today. But we also have a few other groups. We'd like to thank all of our volunteers who go out in the field with us. So a lot of what you're going to be seeing today are not big, well-funded research. There's stuff we do on the weekends. Our friend Don Ashton and everyone else who helps out. Also, yes, I am the second author. I'm the one who gets to get up and talk, hopefully because I'm the one who talks fast and doesn't get as distracted. But the world-renowned expert is here, and you can ask him questions, and I'm going to point at him if you ask me specific stuff. So we're going to go through quickly, right? We're going to introduce what is a western pond turtle. I'm going to summarize some of the current research that's happening, focus a little bit on that research around reservoirs, but our takeaway here is we don't know a lot yet. And then suggest some of the future needs, both in management and research. So first of all, why are we studying this species? Well, they're endemic. They're abundant, especially in some waters. You can find hundreds of individuals in certain sites. They're of high concern, I already stated this, but of high concern to a lot of agencies and also to the public. Plus, they've just been proposed for listing. Comment period is open. We'll come back to that. Now, most of the time, if you have interacted with the western pond turtle, most of these people in this room will have seen one when they came up to bask. And this is behavioral. So they're thermoregulating. You see them up on logs, especially where the water is quite cool. They come up to get warm. And just as a quick note, they are not just bumps on the log. They do have complex social behaviors. Again, something else we'll be happy to tell you about in detail later. Their range is this little narrow strip along the Pacific Ocean, reaching all the way from northern Washington down into Mexico. And there's arguments, extensive arguments about what they should be called, whether there are two species or one species here, and who they're most closely related to. Most likely, most closely related species is on the other side of the planet, the European pond turtle. And though this looks like a huge range, they are disjunct within this because they do require water to feed. So they have to be near water bodies. And so you'll find large swaths of what is covered by that map with no turtles. We go through their life stages. The small ones spend a lot of time hiding because we joke that they are the Oreo cookies of the aquatic ecosystem. Everything wants to eat them. They mostly eat invertebrates. as they spend a long time and a lot of resources building that big, huge armor tank shell, they get predated less. They still have predators. They get predated less, and their diet shifts to be more vegetarian. So as I mentioned, you hardly ever see the little guys because they're busy hiding. And this is a big issue because most of the studies that we're going to talk about are only able to catch adults. They have no idea how many juveniles there are in any of these populations. And that's a problem, and something that we would highlight as, hey, if you're going to go out and estimate how many Western pond turtles are in a population, you should probably try to count the little ones. When they do reach sexual maturity, they're about 120 millimeters carapace length, but that can take a really vastly different amount of time to reach that size. If they're living somewhere really warm where there's a ton of nutrients for them, they can reach adult size in as little as 3 years. If, on the other hand, they're living in a system that's very cold and that doesn't have a ton of nutrients, that can take up to 9 years to reach that same size. Now, right, I mentioned it takes a long time for them to build those big shells. It's a ton of investment. So they do live a very long time once they reach that size. We don't actually know how long they live, but this is our champion. This individual was captured 50 years after she was first marked at 5 years old. And note that in this picture, she was gravid with 9 eggs at the age of 55. Now, I will say, right, we mentioned there are still predators on those large individuals. And over the decades, this is in decades down here, we are finding fewer and fewer of those original marked animals. So they are dropping in the population. The population overall in this particular place is not dropping, but we catch fewer and fewer of those very old individuals the longer time has passed. There are a lot of threats to this species, right? Wouldn't be up for listing if there weren't threats to them. A lot of those threats are to either the eggs or the small ones. But in addition, we have habitat loss, alteration and fragmentation of their habitat, the predation by invasive species. This can get extremely heated. Happy to talk about it later. Competition and the introduction of disease, and climate change, especially worsening droughts and fires. So, status in this moment. The comment period is open on this species. They have been proposed threatened with specific targets for their conservation. And an interesting statement in the proposal is that this proposal has no impact to water use, which is interesting. Again, happy to discuss that, not on the stage. This isn't something that happens quickly, right? Species don't just appear on the endangered species list without a lot of things happening first. So these species have been managed for a long time. There are a lot of statements and information out there. There's a guidance for Oregon native turtles from the Oregon Department of Fisheries and Wildlife. There have been large chunks of funding, which different states used for different research goals. There is a Western Pond Turtle Conservation Coalition that is led by the zoos. A lot of the military installations and lands have had quite a bit of management and research on Western pond turtles on them. And here in Oregon, we have regional native turtle working groups that meet pretty regularly to talk about what's going on with Western pond turtles. All of this information got rolled up into the lovely Western Pond Turtle Handbook a while back. And now that it has passed out of where we have to charge for it every time we give it out, it will be uploaded to SNVB's website for free in the next couple of months. So if you would like to know everything there is to know about Western pond turtles as of like 15 years ago, that will be available shortly. Some of the newer things, Bruce, Germano, Ashton and a couple of other authors are working on a current range-wide comparison of life history, and that is sort of happening right now. So I just told you that lots of people work on them. Everybody's super into them, right? The problem is that a lot of this is weekend warriors. This is people going out on their own time looking for them. This isn't large, focused, well-funded studies. Now, there is a report from 1991 from Holland, which states Western pond turtles have been noted in reservoirs, but in all cases, they consist of small groups of adults, and in no case has a viable population of Western pond turtles been found in any reservoir of any significant size, which would lead you to think, why am I up here? The reason I'm up here is, though Holland is sometimes taken as gospel, if we look at the data, both published and in reports, you can see that there are hundreds of individuals found in large reservoirs. So I would say Holland didn't know what he was talking about. Now, one of the issues here, though, is if you're like, oh, well, I'm going to go look in this reservoir and say this is a good reservoir for pond turtles and this isn't a good reservoir, is that you may be seeing an artifact of where they used to live in the past. So because they're patchily distributed along rivers, if you happen to plonk your reservoir down where there weren't very many turtles, you could have 40 turtles in your reservoir. If you happen to plonk your reservoir down where there were a whole bunch of turtles in the river, you could instantaneously have 105. So this is a good reason to look above and below the reservoir before you just decide, oh, reservoirs are great for turtles. Now, that said, this is a species that relies on quiet waters. They like calm water, either ponds, lakes, or the edges of rivers. They don't like a big, fast rushing river, and reservoirs offer them a lot of literal space to live in. But let's talk about a little bit of the research on those spaces. So we have here whether on the left-hand side, whether the study had a paired river that was undammed, whether they were studying dammed versus undammed. We have whether they looked below the river, whether they looked whether they looked below the reservoir, above the reservoir, in the reservoir, and then before or after the dam either went in or was taken out. And if you notice here, nobody is filling in their row, right? We've got a lot of some above, some below, some in. So we would propose that it would be really important for future studies to start filling in those rows, to look above, below, and in the reservoir if you want to make statements about the reservoir. Now, we've got our little highlighted area up here where it says lost. This is an opportunity that is currently being lost because we have dams coming out right now. So one of the things that we need is we need better statistical rigor and design. We actually need to look before a dam goes in or before a dam comes out. We need to have controls. And if we're looking for density and abundance, we really need to design for that. So that topical dam removal issue here. Dams are coming out on the Klamath. One's already come out. There's a small crew down there, but there's nobody really looking. As they come out, our suggestion would be that when you go from big quiet water to really fast river, to make side channel ponds, right? Really easy to make those in the alluvial plain. Add some woody debris to slow down some of that water, and plants for the juveniles to hide in. We also need to follow turtles for more than just a year, right? If this is an animal that lives for 60 years, managing them for one season isn't a very good plan. We need to protect their uplands. I realize we rushed through some of their natural history, but this is an organism that can be on land for more than 6 months of the year, even though they can only eat in the water. We need to locate and protect their nesting areas. That was a tongue twister. And then we need to enhance the habitat for the hatchlings and juveniles where they're most vulnerable. All right, so what do we know? Do reservoirs favor or reduce turtles? Some minuses. Those big drawdowns might not help them, especially if they're overwintering in that drawdown area. Reservoirs attract humans. Humans like to recreate in reservoirs, and turtles don't like people. But they do create those quiet waters that this species depends on. And some of the reservoirs seem to be places where hundreds of western pond turtles currently live. So evidence says, this isn't something that we have a definitive answer on right now. We don't really know the effect of reservoirs on western pond turtles. Now, the people I've listed so far are not the only stakeholders here. There are lots of other people that are always interested in western pond turtles, and there are regularly lots of meetings about that. So we call for more collaboration, obviously, and for hopefully a more holistic approach when looking at reservoir turtles that focus on above, below, before, and after, etc. And with that, I am ready for questions. Yeah.
Great talk.
Thanks.
What's the potential that reservoirs are acting as ecological traps and setting up sort of source sink dynamics?
Possible. I mean, they can walk around if they really want to, we think. Super awesome, like, research question there is, are they walking around the dams? Or are they stuck, right? Is this a like, oops, we got trapped here and now it like goes up and down 30 meters and nobody can survive the winter? We are still seeing western pond turtles in reservoirs recently. So if we don't think they're getting washed down. So at least some of them, they appear to be surviving great. We don't have data for that.
That's true.
We'd love to have data for that. We don't. Just a second, I gotta check on the list. Yep. Christina talking about coconuts. Yeah.
I have a question for you, Gwen. This is Chrissy, but I can't figure out how to raise my hand.
That's fine.
So when you're seeing them in the reservoirs, are you seeing them in a certain place? Are they closer to the dam? Are they near an inlet? Are they in the middle?
Yeah, they're mostly in some of the reservoirs, and mostly other people's work. They're in the arms, and they're in places where they're not people. I mean, lots of the reservoirs, I mean, there's all the recreation and motor boats and drink, whatever. The turtles are usually where, they're up in the little side arms hiding out. And sometimes where there's a tree falling in the water, the turtles really are tied to cover. They like to hide. They're masters at hiding, and being secretive is real important for their survival. So the turtles are usually where people are not. So even within reservoirs, they're in little pockets around the edges. And if it's real steep, no cover, there's few or no turtles. So they kind of adapt to what's there.
Okay.
Hi, Burries. Are you seeing much interaction with the red ear slider? Can you talk about that a little bit?
Yeah.
So, there are areas near Eugene where they've been removing the red ear sliders, and western pond turtles are doing better in places where they have removed red-eared sliders. Behaviorally, red-eared sliders are much larger as adults, and they seem to take up a lot of the resources that pond turtles would be using. But when we looked for aggressive interactions, right, we showed the like open mouth gestures, like I'm gonna bite you thing. The pond turtles were really aggressive towards each other when the sliders interacted with each other, and the two species did not interact. So as far as we know, they aren't like aggressively interacting, but the red-eared sliders are able to take a lot of resources away from Western pond turtles. Have anything to add?
No, that's true. But overall, the slider is really bad news. It's invasive. They do much better in like in urban areas, and they don't care if people riding around their jet boats, whatever. But they do kind of sort out. But the red-eared slider is mostly urban, but not totally. But the sliders are increasing, and it's a real serious problems. Give credit to ODF&W and also the Corps of Engineers. They've been removing sliders, and it's just like they take them out and they can't, same thing. Yeah, it's just almost impossible to knock them down. But if you do remove them, the Western pond turtles survive, and it's an arms race because the slider produces twice as many eggs as the Western pond turtle. So if we just leave it be, saying, oh, a turtle's a turtle. No, it isn't. The red-eared slider is a very real threat. And there's some good work going on now, and some is starting up in different areas, but the slider is a real threat to the Western pond turtle. And there's also the whole disease issue, which we won't get into, but sliders are not good news.
Awesome.
Thank you very much.
Okay. Good afternoon. Desiree could not make it, so I will keep helping with the presenters. This is the second part of our special session, and it's more focused on reservoir management and monitoring. And it's also a pleasure to me to introduce our next speaker, Greg Taylor. Greg Taylor is with the Army Corps of Engineers, and Greg has been one of the foundational person that helped funding all the basic research that we have in the Willamette River that with Chrissy was showing today, and fundamental for keep doing this. And he will bring a perspective of management of Willamette Reservoir. Welcome.
All right, well, I appreciate it. I especially appreciate an opportunity to speak to a group of scientists today. I have personally been doing a lot of media and talking to a lot of lawyers lately. So I'm looking forward to an opportunity to speak to some scientists. And what I was hoping to talk with you guys about today, and you've heard a lot about the reservoirs already, is some history and a little bit of evolution of how those reservoirs have been managed, and specifically how the water in the reservoirs have been managed. So I'll try to give you a little bit of background today, walk through some of that. And then at the end, I'm going to focus on some of the reservoir operations that we're doing under the injunction. And I'll talk about that. And just give you an example of how different that is. And then, you know, some of the benefits associated with that and some of the impacts. You're going to hear a lot about these. And if you've been reading any of the media recently, the dams, there's been a lot of conversation around some of those things recently. So as folks begin to settle in the Willamette Basin, one thing that was really obvious is they started to look for ways to improve their lives. And in doing that, this river that's right here in our backyard became a big focus, right? And some of my predecessors, I mean, this is going back a long time ago, it's in the 1890s, began to look at the Willamette River and say, wow, can we make this something that can work for us in different ways? And one of the thoughts back then was, well, let's make it more navigable. I mean, look at this, the 1890s. And we had people out there removing huge numbers of snags, wood, gravel, all this stuff. And they do it, and look at the language. And then 10 years later, it's like, well, that was hard. And the river reset itself, right? Big flows, and everything they did was sort of for naught. But just an incredible effort to go out there and make some of these physical changes to the river, and all in sort of this, with this idea that it could make the river better, right? And make their lives better. And so we had this influx of settlers to the Willamette Basin, and they, you know, again, we're thinking in these terms. There's a quote here on this slide, and it says, an urgent need to develop the land and water resources in the Willamette. You know, and that comes from the Oregon State Planning Board. And there was a lot of thought, again, towards looking at this river. How can it be changed or managed in a way that can improve people's lives? And the dams were a big part of that equation, at least for some segment of the public. Not everybody, but a lot of people at that time, I think, were supporters of trying to do something with dams. One of the primary focuses of those early thoughts around the Willamette Project and dams was flooding, you know, and try to reduce some of the harmful impacts of flooding. This is a picture of Portland in the 1890s. I've gone back recently and read some of the testimony that folks gave around the dams. And again, you can get, we're for it, you can get, we're against it. But there really was a lot of testimony by locals and folks that advocated strongly for trying to reduce some of the harmful impacts of flooding. And that really was, at the end of the day, the driver, is these local folks working with congressionals and advocating strongly for a set of dams in the Willamette Basin, something we ultimately called the Willamette Project. So the Willamette Project is this set of 13 dams and reservoirs, but it's also a set of mitigation fish hatcheries, a navigation channel. You saw some of that pictures. Obviously, if you go on the Willamette today, nobody's out there removing snags with these giant boats or doing anything like that. That's not something that goes on today. The lock system and a bank protection program. It's not the river of 250 years ago. It's a highly modified system, right? And that is been driven by these dams and the Willamette Project. For good or for bad, the Willamette Project has a set of authorized purposes. And in some cases, when dams are constructed, there's pretty strict allocations of water, or they're going to operate this way, or they're going to, there's real, the good part of that is, and that's actually how our road projects work down in Southern Oregon. There's some clarity around how to operate. The bad part is you don't have a lot of flexibility. So in the Willamette, we have a lot of flexibility because there's not a lot of sideboards around some of these authorized purposes and how you balance them. And I think if you look over time, you can get a sense for how that balance has changed. And that'll be one of the things I focus on in the talk, is just specific to water management, how some of that balance has changed. And so we do have some flexibility to operate these dams as society starts to value certain things or wants to focus on certain things, to mix and match these things or emphasize some of these authorized purposes more than others. And that's kind of an ongoing evolution and process. So there's been a lot of conversation from the get-go about these dams, and I've kind of alluded to this. I've done multiple news articles just in the last few weeks. There was one that came out just a few days ago on the turbidity associated with the drawdown at Green Peter. You might have read that. We had one a few weeks prior to that ProPublica did on kind of questioning our plan around operating the dams and salmon. I mean, but this is the same conversation we've been having for 100 years. Really. I mean, seriously. I mean, we go back to what William Finley said. What does that say? I'm having a little hard time seeing that, but basically it says we're going to have dams and no salmon. That was 100 years ago. And so we're having this ongoing conversation about these dams, about their value to society, how they should be operated, how they should be managed, what should be emphasized, what purpose should really, drive the bus. And that's the way it should be. We should continue to be talking about these, making decisions about how to best operate them. And I guess from where I sit as one of the people that gets to steward some of this, I hope that it reflects what society wants. Maybe it does, maybe it doesn't. But ideally, I think how we're managing the system would reflect societal values. There's a quick timeline. You've seen this already a few times today. These facilities were authorized in the late 30s. The first construction or projects were in place in the early 40s. And then there's kind of this period of dam building in this area from 41 through 69. So all 13 dams and reservoirs were in place over that particular time frame. As I kind of walk through the rest of this talk and then kind of start comparing some of the water management aspects, that I want you to keep in mind some different eras and some different policies that drove, from my perspective, different water management in the basin. And so I just mentioned, obviously, the dam construction era, 41 through 69, but I've defined an area kind of post-construction from that point through about 1999. And the reason I did is that if you go back and look at how the dams were operated, they really operated pretty tight to the rule curves, and we're going to talk about the rule curves next. They're fundamental. The rule curves that each of the dams have. I mean, they're authorized as part of the project, and they really trace those quite closely, by and large, in this era. That all changed in 1999 with the listing of Upper Willamette River Spring Chinook and Upper Willamette River Steelhead. And so, that's a different era. We had now things like main stem flow targets, tributary targets, and the practical effect of that, and you'll see it in a little bit here, was that more water was going downstream instead of into the reservoirs. And so that was a real driver. And then the most recent era is we're under an injunction, and we have a judge's final order that drives our operations. And so that's another era, and you'll see all of these as we kind of walk through those, and you can compare and see the evolution that's occurred over time in our water management. All right, so I mentioned this. You got to know what this is. The rest of this isn't going to make any sense. So what this is a water control diagram. Each dam has one of these. And each one of these is specific to that particular dam. So on the x-axis is date. On the y is elevation. Generically speaking, these dams are generally at their lowest in the winter, okay? So December and January. As flood risk starts to diminish, they start to refill, starting on February 1st, typically. They go through a refill process, so they're capturing more water than is coming in, right? So we've got less water going out than what's coming in, and we accumulate over the entire system about 1.6 million acre feet. They're held relatively high for the summer when there's very little risk to flooding, and then they're drawn back down in the fall and the winter. And that's classically how they're operated, okay? Got to have that understanding. That's just how they're managed. Now, there's one really key point here too. We have flexibility to operate these dams kind of in one direction. And the one direction is to be below the rule curve versus above it. The only time we can be above that rule curve on a particular date is if we're literally capturing water to keep control points downstream of the dams below flood levels, essentially. So if we're taking a flood damage reduction action, then we're going to capture water, allow the reservoir to go up over the rule curve. But other than that, we can't be cheating or above that rule curve. On the flip side, we can keep this down. We can keep it down. We don't have to store the water per se. We obviously do. And there's a lot of value in storing the water for a lot of different things that are done with the dams. But we do have that flexibility. So important to know that as well. So I'm going to try to detangle this mess. This mess is the operation of Lookout Point Dam over the last 20 some odd years. And how we're going to look at this is we're going to take the reservoir elevation, okay? We're going to look at some different eras, and then we're going to contrast that with the rule curve. And we're going to use that as our metric to sort of look at water management, right? And kind of the idea here is as the reservoir elevations have changed over time, that's reflective of different water management in that we're either putting more water downstream or less water downstream and keeping the pool higher by doing that, or drawing the pool down. So, okay. And yeah, I didn't expressly mention this, but the thing that has driven the biggest changes in how we've managed water, bar none, is the listing of endangered species in 1999. That is absolutely 100% the thing that has driven differential water management in the basin. And it still is doing that today. So really fundamental. So here's Lookout Point. To orient you, this black line, that is the rule curve. And as I mentioned, that is essentially specific to Lookout Point. So on a particular date, you would be on that black line at that elevation if you are managing the reservoir exactly consistently with the rule curve that we have for that site. You can see in the pre-ESA kind of post-construction era, the red line. And I chose 15 years to kind of accumulate the reservoir elevations over that time and just kind of smooth them out. And what you can see is that largely tracks, by and large, with the rule curve. You know, pool comes up, stays up, drawing back down in the fall. Contrast that with the green line. And the green line is post-listing, an additional 15 years at Lookout Point. And you can see there's some changes there. And you know, the pools don't come up by and large as high. They don't stay up as long, and they come down faster. And that is driven by more water being released downstream than we did pre-ESA listing. Okay. And you could go back and you can take, you know, these elevations and convert them to acre feet of storage. And you could also look at how that kind of manifests itself downstream as actual flow during specific periods. But we are not accumulating as much storage, generally speaking, and we're not holding that storage as long as we did per the rule curve. If you look at other projects, here's Green Peter, same thing. You know, Green Peter, you're seeing that same trend, really closely following that water control diagram, pre-ESA, post-ESA, a big change. And then I didn't mention it on Lookout, but under the injunction that we have from a federal judge from 2021 on, at some sites, we're doing these drawdown operations. And that's what this blue line signifies. I don't think we need statistics to say that's different than how we used to do things, right? I mean, sometimes a picture just says 1,000 words. That's different. It's radically different. And in this case, at Green Peter, and we're gonna go through Green Peter specifically. You know, we're going down 142 feet below our normal winter set point. This reservoir fluctuates about 100 feet annually here. So, you know, Chrissy was talking about some of the reservoir fluctuation. I mean, we're talking a couple 100 feet or better annually that we're doing with these drawdowns. Still a residual pool here, multi-mile long reservoir. We'll talk about that. These are departures from how things used to be done. And I just want to paint that picture. So if you didn't have an understanding of how this has evolved over time, there've been some significant changes. And that's not true of every location. Okay, so, and that's one of the things to talk through today is each site is a little bit different. Each dam is different. You know, Detroit, you can see that same trend that we just talked about at Detroit, but it's not as apparent. There's reasons for that. You know, Lookout Point and Green Peter are higher priority for augmentation downstream, so they're being pulled on harder. Detroit has infrastructure high in a reservoir that we like to use for downstream fish passage and for spill. So it's beneficial to stay up there a little bit higher in elevation to reach that. So all sites are not the same, but there's still some differences there from the historic. All right, so we're going to transition here. Hopefully, you just got a pretty good sense of how we've kind of managed water differently over time. What I want to do now is just walk you through some specifics of before we ever release a drop of water, what are some of the things that are on the table that we're thinking about and have to manage to? And then after we do that, we're going to spend some time talking about some specific examples so I can give you some clarity around that.
Greg, could you just explain that injunction? Why that?
Yeah, I can do it now, or I can do it as a question. We were found to have violated ESA is sort of the short answer. We had in 2008, we signed a biological opinion that had a 15-year implementation time frame. In 2019, we were sued under ESA. The basic the groups that brought the lawsuit said that we hadn't done what we were required to do under that biological opinion. The judge agreed. And so he found remedy, and he made a decision that we had violated ESA, and then the plaintiffs had remedy. And so the remedy in this case was going back and looking hard at those operations and whether there could be some changes. And those curves that you're seeing under the injunction, or the changes in the rule curves, were part of that remedy. So a very different operation than what we had done in the past. The idea being that that would be beneficial for fish, and we'll talk about that. So did that answer? Okay. So Mother Nature is still in charge of our highly modified system. And, when we have, this all starts, the whole system starts with the inputs from Mother Nature, and specifically the water. It's very different if you get 7.5 million acre feet that run off at Salem than 2.5. I mean, I'll tell you that's 5 million acre feet of water. You get different outcomes when you're, depending on what Mother Nature provides. And I give you those numbers, and if you don't believe them, they happened. Those two extremes happened in 21 and 22, back to back. 21, we had about 2.5 million acre feet run off at Salem, and in 22, we had 7.5 million. So we get very different outcomes. You get very different reservoir levels, you get very different releases of water downstream, but it all starts there. You're managing to the rule curve. That's why we talked about the rule curves. The rule curve is a real, really drives what you can do. But you're also managing for a lot of things downstream. We have downstream control points. I mean, I put that in there as a bullet. There's 32 downstream control points. It's not like there's one, there's 32 that we are trying to manage to make sure that we don't exceed those and flood people. A whole system of those things that we're looking at. We got main stem flow targets. We got tributary flow targets. We got minimum flow requirements. At the dams themselves, we're managing hydropower and outage schedules associated with that. We've got all kinds of special operations we're doing these days to support research, et cetera, et cetera. There's just a lot of balls that are in the air, different types of operations we're doing for spill, for temperature management under the injunction. It's not to paint a picture of, woe is the core. It's to give you some realistic sense of a lot of the balls that are being juggled. And, all that in the context of a fish, right? What are the fish doing? A ton of things. Different species do different things, right? Winter steelhead spawn in the late spring. Spring Chinook spawn in the fall. What does that mean? Well, that means there's eggs in the gravel at certain times for certain species. That means there's different life history patterns that are playing out on the ground out there. All that matters. And all that has to be taken into consideration when you're managing this system. So you're doing all this stuff at the dam, but then you're also thinking about these endangered species and all the things that they're trying to, their aspects of their life history that are trying to take place out on the ground and in the rivers below the dams. So there's a lot to it. There's a lot to it. All right, so we're going to walk through a real world example of how you operate a dam to try to help something. And the something's going to be an ESA-listed fish. So this has been my life for 22 years. So you get a little insight here. And I want to just walk you through something that you can read about in the paper this week, which is the drawdown at Green Peter. And I'm going to walk through the good, I'm going to walk through the bad, and I'm going to kind of walk you through how you design something like this, and then you know, kind of work your way through it. So when you do one of these operations at the dam, the place you start is, well, what does that dam have as infrastructure? I mean, the dam was designed 100 years ago. It wasn't designed last week with ESA listed fish in mind. It was designed 100 years ago. In some cases, fish were a thought. Some cases they weren't. A lot of cases they weren't. And so you have to look and see what's available. This is a picture of Lookout Point. It's our largest storage reservoir. It has spillway gates high up in the reservoir. Okay, well, that's good. You can use that to spill water in the spring when the reservoir is full, and we do, and that's helpful. But not all the reservoirs have this. Some of them have penstocks, turbines. Some of them don't. Some of them have regulating outlets deep in the reservoir. Some of them have regulating outlets that are at 2 different elevations. At Lookout Point, they're at 1 elevation. At Green Peter, they're at 1 elevation. Happens to be very deep in the reservoirs. So you really start with your infrastructure, and specifically what tools being these different gates that you have to pass water downstream. That's where this all starts. And, you know, in the short term, the things that we can do for fish, they really are operational. We're not going to be building large scale structures in the short term. So all the things that we're doing under this injunction and in the near term are really these operational changes using the dams, using the infrastructure, operating the dam differently, you know, changing the elevation of the reservoir, using a regulating outlet. So those are the things that we'll focus on. Under the injunction, these things kind of fall into 3 buckets. They're really spill. Okay, you can see this is at Detroit. We're using spill. What's called the delayed refill. So we're not filling the pool up as much. And the reason you do that is to try to keep fish closer to an outlet structure, essentially. And then drawdown. And so we'll focus on this drawdown at Green Peter. All right, there's one slide that you pay attention to in this entire presentation. When you do these operations at these dams to help fish, they're typically in the arena of flow, changes in flow, changes in water quality, or fish passage. Always have impact, and then there's always some benefit too. And the trick is to understand what those impacts are, what those benefits are, and make that trade-off, and understand that there's going to be trade-offs between species that might both be listed on the Endangered Species Act, even within a species, their life history, you know, the incubating eggs in the gravel versus the adult versus, and then obviously our authorized purposes, right? If we're keeping more water in reservoir or less water in reservoir, we can really have trade-offs that we're making between our authorized purposes. So no free lunch. There's not an operation out there that exists where you implement it, it's all positive for everything we do. It doesn't exist. They don't exist. So you really got to go through and understand these trade-offs, and then work together regionally to figure out what the best approach is. All right, Green Peter. So at Green Peter, here's the roll curve. And you can see this blue line. That is the drawdown that we're doing under the injunction. I mentioned it before, it's 142 feet below our normal set point in the winter. So the way it looks, this light blue in this picture is Green Peter when it's full. This medium blue, let's call it, is Green Peter when it's at its normal winter set point, okay, drawn down for floods. And the dark blue is Green Peter after this drawdown. And if you go out there this afternoon, you can see it just like this. It's down. This is roughly 3 miles from this point to the dam, okay? So there's still a reservoir there. It's a lot narrower, it's a lot shallower, and it's a lot dirtier than it was a month ago, okay? But there's still a reservoir. That is a difference than a full stream bed drawdown like we do at Fall Creek, where we take it all the way to stream bed. But it's still a radical departure. Why is it where it is? Well, the reason is we've got to access our regulating outlets, okay? And our regulating outlets at Green Peter are very, very deep. And so that's what's really driving this. Let me walk through this a little more conceptually. So here's what we're trying to do on the biological side. This is the good. This is the benefit piece, okay? So I want to walk you through that. So when the reservoir's full, we have juvenile spring Chinook salmon. They live in the reservoir. You've heard that today several times. They're surface oriented by and large. That's where they live, you know, in the top 20, 30 feet of the water column, sometimes deeper in the summer, but they're relatively surface oriented. So when the reservoir is full, we have spillway gates at Green Peter. We can open those and we got a surface route. That's a safe route for fish. It's not a turbine unit. It's not, it's just a gate. And so fish can go out through that. And we're doing that under the injunction. But as you start to draw the pool down a little bit, now you're left with two other choices. Right? Now your two choices are the turbine or the regulating outlet. That's all you have. I mean, that's where the water is, can go. And so at this site with this turbine, it's a high head Francis unit. They have really poor survival. I mean, that's just the reality. So the turbine unit is not a good option. And even at minimum conservation pool, it's still quite deep in the reservoir and actually pretty hard for fish to find. So the drawdown, what it's doing is we're going to shut the unit off. We're not going to use the unit. And we're bringing the pool down so that the fish can get within striking distance of the regulating outlet, which is the safe route through the dam. Now, it just so happens that we need to pull the pool down 142 feet to do that, we think. And usually the striking distance for these fish is around under 50 feet from the water surface to whatever intake that you have. They tend to find those outlets well. The shallower, the better. So in this case, we're going within about 25 feet of that regulating outlet. And that's what's hopefully going to pass these fish out. We're going to try to get, it's no more complicated than this. We're going to try to get more fish out at a higher survival rate. Period. I mean, we'll make it way more complicated than that, but that's really what we're trying to do at the end of the day. That's what we can do with the existing infrastructure, and that's what the drawdown is all about. Where we've done this, I should have brought that down. So yeah, so there you go. So as we get the pool down, they have the regulating outlet, the fish can get out. That's the way this conceptually works. We've done this. We do this at Fall Creek. At Fall Creek, as we pull the pool down, there we go all the way to stream bed. The red here, or the blue here, is the reservoir elevation. The red is juvenile Chinook passage. Doesn't take a rocket scientist to tell you that more fish come out of that project as the reservoir elevation goes down. This is real data. So yeah, the fish move as the depth to the intake narrows up, more of them find their way out. So this can work, does work. There are trade-offs. There are impacts. This gray line is the water temperatures below Foster Dam in the South Santiam River. Understand that the area below Foster is one of the highest density spawning areas that we have for spring Chinook in the basin. Okay, so we're up there doing this good thing at Green Peter, getting the fish out, you know. That gray line is not good. That's 2023. And this year is the first year that we've implemented this drawdown. What we're seeing is very high water temperatures in the fall associated with the drawdown. I've got some ideas about how to make that better, but that's not going to be in place this year. The thing I caution is that in thinking about trade-offs, make sure we understand what we're trading off. If you know incubation like I do for spring Chinook salmon, 62 degrees after all the eggs in the gravel is scary. It's scary as hell. You could lose all those eggs. Over at Dexter Dam and in the Middle Fork Willamette below the dam, that's always been true that we exceed these temperatures, and we know for a fact that nobody is successful spawning below those areas. So we've had an area that is, well, fish do spawn successfully, but we're getting this result that's not great. It's not what we want to see, but it's part of it. And again, this is understanding that full picture of, you know, you take an action, how does that action sort of manifest itself on the ground, and what are the trade-offs that sort of result? This was a picture that was taken last week. That's the South Santiam River on one side, the North Santiam on the other. They look different. Why do they look different? Well, because one of them has really turbid water from the reservoir being drawn down. One of them doesn't. When you do a drawdown, you have to let that river recut a channel through 50, 70, 80, 100 years of sediment that's accumulated in whatever dam you're operating. And that's what's going on here. Now, was it unexpected? No. Knew it was going to happen. Is it problematic biologically, socially? Hell yeah, it is. I can tell you just about everybody in Springfield has my phone number, and they're calling, or in Sweet Home, and they're calling me. They don't like it. They don't like it for a whole bunch of reasons. So it's something to manage. It's something to understand. You got to know that these, now, will it get better? Absolutely. We saw this at Fall Creek, you know, the first years were the worst when we drew all the way down. Over time, as that river can move some of the sediment, theoretically, we're not going to see as much of this turbidity. It'll start later. Hopefully the duration and magnitude will come down. It'll get better, but it's real. And it is, I don't believe this is lethal to fish based on our work at Cougar when we did something similar to this. But I also know that we put fish in live cars at that same project. And during a similar type turbidity event, we did see some gill irritation and damage on fish. No deaths, but we saw some impacts. And so just got to be intellectually honest about that, got to understand it and factor it in as you move forward. And, you know, this just gives you a sense of some of the turbidity we're seeing down there. We're obviously hearing from the public about it, and it's just part of this, and something that we're going to work through as we manage these reservoirs. So there's Green Peter. That's just one example. We're doing this at Lookout. We're doing it at Fall Creek. We're doing some version of this at Cougar. We're doing a million different types of operations under the injunction. I don't have time to talk about all those today, but hopefully this gives you some flavor for how we've operated these reservoirs differently in the last 50 to 70 years. Some of the new things that we're doing that are more visible to the public. And just, you know, there have been changes. You know, we've made some significant changes. But as we do those things, you know, there's trade-offs, and that'll be something that we have to work regionally on together. The system will continue to evolve. I mean, it just will. You know, we're going to keep working all together on this to try to make improvements that hopefully reflect societal values. A lot of those are driven by ESA right now. A lot of those are driven by ESA. So I appreciate the opportunity. I appreciate your time, and hopefully you have some questions I can answer. Thanks.
We have time for one short question, and then we have the coffee break. We have more time for discussion.
You.
Testing. Okay, good. Have there been any impacts on drinking water downstream with the spikes in turbidity in terms of treatment or having to switch water sources?
So my Monday morning, a team of us met with the cities of Sweet Home, Albany, Lebanon. Next Monday, we're meeting with Lowell and some others. I want to make sure I get this right. The cities knew that some turbidity was coming. I don't think they fully understood the duration, the magnitude. It's a significant, I'd characterize it as an operations and maintenance issue. They can do it, but they're not staffed up to do it right now. And yes, we heard very, very loudly from them on Monday about some challenges they were having. So they're trying to work through those. And we don't, in our case, have a lot we can do to help them, unfortunately. But there's some challenges. The water's safe. Water's safe. They're treating water. But they have some significant operations and maintenance issues that are challenges, for sure. All right, catch me at the break. Appreciate it. Thank you.
Thank you, Greg. A really kind of a realistic view of what's going on in situ. So next presenter is Brett Vanden Heuvel, talking about time of change of Upper Willamette River reservoirs.
All right, thank you. Hi, everybody. I'm Brett Vanden Heuvel. I'm with the Confederated Tribes of Grand Ronde. Really happy to be here. It's been a fascinating morning, learning about a lot of the science that's going on in the reservoirs. I'm going to give more of a policy talk. My background, I'm an environmental lawyer. And it's great to follow Greg because that set up a lot of this conversation really well. So I also want to introduce Grand Ronde Tribal Councilwoman Kathleen George is here today. And Kathleen has been one of the champions at the tribe of setting up the Salmon Strength Initiative that I work for, with the goal of preventing extinction and restoring these species. So I just want to start with Willamette Basin salmon are essential to the culture and the lifeways of Grand Ronde people. This is a map of the homelands of the Grand Ronde, and there were 6 treaties signed in 1854 and 1855 that ceded lands to the United States. But as you can see, the homelands of the Grand Ronde include the entire Willamette Valley and beyond, where all of these projects are located. And the tribe is committed to bringing these fish back to healthy populations. So we've talked a lot about the 2 ESA-listed species. According to NOAA Fisheries, the Upper Willamette River Chinook has a very high likelihood of extinction in most of the subbasins. So just to put a finer point on that, there's about 10,000 wild Chinook that return each year on average. That's about 97% decrease from historic estimates. The steelhead would be in about the same boat. There's a little over 3,000 that return each year, a fraction of the historic populations. And we have an opportunity now. I think the exciting part is things are changing. Things are changing, and we have an opportunity right now to prevent extinction, which again is very likely to happen unless we make pretty significant changes, and to restore these populations. And I think one of the really exciting things, some of these fish, I mean, they are outplanted above the dams. The challenge is getting the juveniles back down, but that habitat above the dams, there's a lot of it. Most of it's on national forest land, and it's not requiring a lot of restoration like many, of our rivers. So if we can get the fish there, I think there's really exciting opportunities. Okay, I'm going to dig a little bit into this timeline, and I guess this is what Greg would be saying is the sort of post the ESA listing in the injunction era. I put it all together here. So in 1999, these 2, the Chinook, the wild Chinook, and the wild steelhead were listed. About 10 years later, NOAA Fisheries BIOP said that the operation of the dams is jeopardizing the existence of these fish. And so they required certain actions. And, over the next 10 years, the Corps did not fulfill most of those obligations. And I want to be super clear that, the employees at the Corps, are doing incredible work, are working very hard, are taking many, many actions to improve salmon. It's just some policy decisions, some fundamental policy decisions that aren't necessarily being made on the ground here that are greatly constraining how successful those efforts can be. So some environmental groups challenged the Corps, and a couple of years later, an opinion came out from a judge and the injunction that Greg mentioned. So I just want to read this little quote. Far short of moving towards recovery, the Corps is pushing Upper Willamette River Chinook and steelhead even closer to the brink of extinction. I've worked for a federal judge. I know they are loath to tell agencies what to do. They're loath to issue injunctions. And they heard from lots of experts on lots of different sides. And, the experts, the biologists hired by the plaintiffs, the biologists from the state of Oregon, federal biologists, retired and otherwise, helped to craft some of these measures. So it's not the judge, saying, I think this should happen. This was seen as necessary to prevent the extinction of these species. The kind of third to last bullet here, the other thing that was happening sort of simultaneously is Congress was acting. So WRDA, the Water Resources Development Act, is a a bill that directs the Army Corps operations. And so in 2022, Congress asked the Corps to evaluate the deauthorization of hydropower for some or all of the Willamette Valley system. And in 2020, prior to that, they asked them to do the same thing for Cougar and Detroit dams. And so Congress is asking questions, does it make sense asking the Corps to conduct the studies, a report to look at whether they should remove hydropower from these projects? Put it very, very clear, not talking about dam removal, right? We're talking about not spinning the turbines for hydropower. The dams would stay in place, and that rule curve, the change of filling up the reservoir may not be necessary anymore for hydropower. So if the dams would stay in place, the various outlets might be open, and if there is flood risk, they could close those, fill up the reservoirs to the extent needed. So right now, just last year, the Corps issued a draft environmental impact statement, and in 2024, there's a deadline from the Corps to issue a final operating plan for the dams, environmental impact statement as well, and a new BIOP too. So that's sort of a little bit deeper dive into where we are in sort of the regulatory process. So these are changes. Yeah, Greg did a great job explaining that. And I want to touch on one other thing that hasn't really been brought up today as much as the economics and the power production for the dams. And so the dams were designed, the primary purpose was flood control. Hydropower has never been a primary purpose. And I just have a picture here of Green Peter as an example. Green Peter produces on average about 30 megawatts per year. That's about as much power as 10 wind turbines. It's not much. And then here's a table that has the annual power production for the dams, and the total down there is 187. That includes Dorina, which is not a core dam, so that the average is about 184 megawatts. That is 2% of the Federal Columbia River power system. So the Willamette produces about 2% of the federal system on the Columbia. To put 184 megawatts total, this is total into perspective, the nameplate capacity for John Day is about 2,500 megawatts. Grand Coulee is over 6,000 megawatts. And that's nameplate, so that's a little higher than this. But, there's some big solar projects proposed that are 400 megawatts, one in Central Oregon. There's one in Morrow County, it's proposed, it's 500 megawatts. So I'm not saying 184 megawatts is a small amount of power, but relative to some of the new things that are coming on, relative to the Columbia River system, and relative to the extreme impact that it's having on salmon, just want to put in perspective, this is not, this is 2% of the FCR RPS. The other important part I think about hydropower here is this first line, which is, it came from the environmental impact statement, and it's talking about net present value, but there would be a $939 million reduction in net present value to negative $714 million for the Corps' preferred alternative. So over the next 30 years, if we go with what the Corps is proposing as a preferred alternative, the dams would, the costs would outweigh the revenue by $939 million. The dams are losing money, would lose a substantial amount of money to produce hydropower. And then the long quote at the bottom from Bonneville Power's fact sheet, basically saying that, it's 30, it describes the cost a little bit, and this is in 2019, and saying additional fish mitigation measures are going to make the cost more expensive. We certainly have those already. So in short, we have these dams are producing the hydropower about 2% of the hydropower for the Columbia system. And it's roughly 3 times more expensive than other power from other dams. I say all this to, put in perspective, the management of the dams it's for multiple purposes. It's primarily for flood control, but that rule curve, the refilling of that rule curve, the primary reason we're doing that is to produce hydropower. And the numbers are starting to make people ask why. Why? We can look at a lot of the science of the reservoirs, but I think a fundamental question right now is, why are we having these why are we following so closely to the rule curve that's designed for hydropower production when it's very expensive and it's not a huge amount of power? I'm oversimplifying things a lot to kind of go quickly here, but this led to a recent article, one of which that Greg mentioned, killing salmon to lose money, a costly, questionable plan on the Willamette. One of the things that has proven to work, and I know Greg was part of the early work on this, making this happen at Fall Creek long before the injunction, is drawing down the reservoirs. And I don't need to spend much time on this. You understand this, that you're drawing down, you're able to get the juvenile salmon out quicker. There's less, it's not a stagnant reservoir, it's a moving river. There's probably less predation. We heard about parasites earlier. Maybe it'd be less of a problem for the copepods if you had flowing rivers more often. So we've seen this as successful. In fact, some of the Corps' work at Fall Creek showed a 10 times the number of adults returned from that drawdown compared to a fuller reservoir. So drawdown, we think deep drawdown is good for salmon. It's good for flood risk mitigation, because if you need to, empty reservoirs can fill up. And yes, each dam is unique. Yes, there's impacts. Yes, it's creating muddy water. It's creating turbid water that, is people are going to have some concerns with. Yes, we need to develop solutions for station power, for water quality issues, and perhaps build some additional infrastructure. But I'm 100% convinced that we can overcome these challenges if there is policy from Corps leadership that is focused on prioritizing salmon recovery as a primary goal here. If we try to do a little bit of everything, that has failed. That's failed in the past. It'll probably continue to fail in the future. So one of the recommendations that the Grand Ronde tribe has is, one, to prioritize salmon recovery over hydropower, and to also look very closely at the concept of deauthorizing hydropower. One of the things that, talking to a lot of folks at the Corps, that their policy people feel constrained by is that they have to produce hydropower. Whether that's true or not, or how much they have to produce is kind of an open question. But a way to solve that problem would be to ask Congress to take that out of the uses for the dams. And that would free up the Corps to have a lot more flexibility in how they're operating the dams. And so right now, historically, maybe drawdown would be for a couple weeks. The injunction is certainly making it longer. Maybe it's 4 weeks or 6 weeks. Thinking into the future, if hydropower is not an authorized use, some of the dams the reservoirs could be drawn down much, much longer. Perhaps the run of river for certain dams. Perhaps you're leaving them down letting vegetation grow along the banks and, filling them up as you need to for flood risk mitigation. So not pretending to have all of the answers of how that works. One of the other things that we've asked the Corps to do is to look at a no power alternative as part of their environmental impact statement. The draft, they did not do that. We think that was unfortunate when they're doing a 700-page study or whatever it was. This seems like a very real possibility into the future. And so we're currently asking the Corps to take a harder look at this. What does that look like? Evaluate those impacts, because there are very real trade-offs, benefits, and harms by doing, all these different things, as was explained so well earlier. But we think the Corps has that in their power, and they should be considering that right now. So I'll wrap up with just this photo of, this is way up in the Upper Willamette system, and our hope and Grand Ron's commitment is that we're making decisions now that will allow the salmon to continue to return and reach some of these upper regions where there is such important and valuable habitat.
One question online. You can just see this one.
Okay, we'll go through these quick. Thanks. Let's see. Seeing a comment there. See if I can find a question. Does this seem accurate? For conventional generators, such as a coal plant, a megawatt capacity will produce electricity that equates to the same amount of electricity consumed by 400 to 900 homes a year. So 187 megawatts equals 75,000 home to 169,000 homes per year. I don't know those numbers exactly, so I'm not sure on that one. I would need to look those up. But I think what they're trying to say is it would be a lot of homes, and I would say, yes, that's great. There's also other ways to produce electricity that to power those homes.
Any question from the room? We have a little bit of time. Can you use the microphone, please?
I was curious about the legislation that might be in the works for more of like the deauthorization of reservoirs and hydropower, and kind of what's in the works for that.
Yeah, right now we're still based on, so the Corps was supposed to complete a study from the 2020 legislation. I've heard it's done. They haven't delivered it to Congress yet. The broader study, the Corps actually just did listening session yesterday on this disposition study, or disposing of the federal asset of hydropower. So that's happening right now. The Corps is doing hopefully a deep dive on that. Into the future, I think we need to see what happens with these existing studies. But I will say, unlike some other dam and conservation issues, there is a broad interest here. Bonneville Power, that manages that markets the power from this system, I think they're very skeptical about making money in the Willamette based on some of these numbers, some of the trade associations as well. So there's sort of a broad group of people looking at this that have some concerns about continued, status quo as it is for the hydropower. So I can't speak to the exact upcoming legislation, but I expect there will be some additional language in the 2024 WRDA. The WRDA is every 2 years. Yeah.
Thank you very much. Right. Right, our next speaker, Dalton Hance with the USGS, talking about multidisciplinary work in reservoirs.
Thanks, Yvonne. I just want to real quick thank Greg and Brett for sort of leading me off and teeing this off, because it's really nice to hear the management perspective and kind of a policy perspective. I'll just say one of the great things about my job and working for the agency that I do is that I don't have to manage anything, and I don't make any policy decisions. In fact, we can't even make recommendations on the policy decisions. But what I want to do is bring us back to some of the science from this morning and really talk about the science of fish passage, because that's really what we're talking about: managing these dams, making policy at these dams for fish passage. And so kind of bring us back to what do we know and what don't we know about fish passage. So this should be familiar for most folks by now. Today, what we're talking about is the Willamette Valley project. Primary authorization is for flood control with several other secondary priorities. Management for ESA-listed salmonids, particularly Willamette Valley Spring Chinook, is rising in those priorities to the point where it's now probably the second priority for managing these dams. So this is the typical Chinook salmon life cycle. This should be familiar for most folks, but the idea is that the juveniles are born in fresh water, they rear for a time, and then migrate down rivers to the ocean where they grow big and then come back, right? So that's your typical Chinook salmon life cycle. So in the middle fork of the Willamette, the life cycle involves two other steps, really. So the adults can't pass Dexter Dam. They need to be loaded onto trucks and taken upstream where they spawn. They produce juveniles, which then have to get past Lookout Point Dam and Dexter Dam. And as Greg pointed out, there's really only so many ways to get these fish past the dams. And so that's either through the spillways, through those turbine intakes, and through those regulating outlets. And just know that there's different water elevations. So I really want to thank Greg for bringing up the rule curve, because I'm going to come back to that a little later in this talk here. So this is the goal set by policymakers, by National Marine Fishery Service, and by the recent court decision with the injunction. The goal is to establish self-sustaining Chinook salmon populations upstream of the Willamette Valley Project dams. That requires, and I quote, safe and effective juvenile fish passage for all size classes of juvenile migrants. So that's what we're talking about, juvenile fish passage. But what I want to focus in on is how, when, and what are the other effects of those actions? What do we know and what don't we know? And so this is going to be reviewing some of the stuff from this morning. What do we know about juvenile Chinook salmon in Lookout Point Reservoir? So this is from Schroeder et al. 2015, mostly based off of Mackenzie River. These are the migratory pathways for Willamette Valley Spring Chinook salmon. And I think Chris had a version of this in his talk this morning. But basically the idea is that there's three main pathways when you're going from the headwater to the reservoir. There's what these guys called movers or stayers, but really what we're talking about is fry migrants that enter in the spring, fall migrants that are sub-yearlings, and then yearlings, which will come out of the headwaters the following spring. And then they can pass Willamette Falls at different times. So we can have autumn smolts or yearling smolts. So we see this in the middle fork of the Willamette. We also see evidence of these three different migratory pathways into Lookout Point. Spring sub-yearlings, which are small fish, fry, that's in that sort of purple circle there, entering from March through about June in 2016. This is from the North Fork of the Middle Fork smolt trap data. And then we have fall sub-yearlings and then spring yearlings. I just want to point out that this is data coming from a smolt trap. The observed number is not abundance. A smolt trap does not capture every fish. It changes with water conditions. It changes as fish get bigger. You can't even really look at the relative numbers. We actually don't know what the relative contribution of these different pathways is to fish in Lookout Point and passing Lookout Point. But I bolded the fry migrants because we're going to talk about them a little bit more in detail because they're probably the most affected by operations in Lookout Point. So
once they get into Lookout Point, what do we know about their distribution? And so again, this is these fry migrants. What we're looking at here is the longitudinal distribution through the reservoir over time in 2 different years. On the left is 2017. That was a wet, cold year, one of the wettest and coldest on record. There was actually spill this year, cold water, high water levels. What we saw in that year, particularly in the spring, in the very first year, very first part of the year in May, fish were sort of evenly distributed throughout the reservoir. And then in June and July, there was more fish close to the dam. And then they kind of spread it out again. And then by September, October, they're clustering close to the dam. In 2018, we didn't see that same thing in June and July of fish being right up at the dam. There wasn't spill in that year, but what we saw was a sort of, more at the head of reservoir, then more distributed throughout. And then by September, October, getting closer to the dam. And this is from GillNet data, capturing fish in the dam. Depth distribution. In the spring, when water temperatures are cold, they are higher in elevation. That's where you're finding them. By August and September in these years, particularly in 2018, they were a lot deeper, up to 30 meters deep. And really where we found them was in this band of temperatures between 12 degrees Celsius and 15 degrees Celsius. And Chrissy talked about that this morning, this idea that there's sort of an ideal sort of temperature for them to metabolize, but they will go higher up to find food. And we know that there are rich food resources in these reservoirs for those fish that enter in the early spring as fry. Again, Chrissy talked about this morning, so I'm not going to spend a lot of time here. We also talked about growth this morning. So we know compared to the fish that are the stayers, those sub-yearling fall migrants, fish that enter in the spring as fry have much greater opportunities for growth. And so that left figure is just kind of comparing the two. It's from, I think this is from Monzik, 2014. So there's a growth potential there. But there's also a predation potential. There are numerous non-native predator species in Lookout Point Reservoir, and we know that they do eat those fry. So that's this is another one of Chrissy's papers on the left there. The red is basically evidence that they were eating this marine-derived nutrients, which can only have come from the salmon fry. And then we also talked about the copepods this morning. And so this is sort of an emerging issue in our awareness. It's not necessarily an emerging issue for the species, but we do know that the fry are probably the most affected by this. So again, it's a sort of review. But what I do want to talk about is that temperature and depth affect food and parasites, growth and survival. So it's really about where is the cold water, where is the warm water, and where the depth is. So they are able to grow because they're able to take advantage of having cold water. It's there for them to migrate down to. Food resources are where the warm water is, but then that also passes them through where the parasites are. So it's a story of temperature and depth. And really, that's function of dam operations. There's a lot of things that can affect water temperatures and the depth at which those water temperatures are found. The lake level, so the inflows and the outflows, the temperature profile, the kind of year it is, whether it's like I showed you 2017 and 2018, one was a wet, cold year, one was a warmer, drier year. So there's a lot of things that can affect these things. Mother Nature is one thing. Dam operations are another thing that can change this profile. But we do know that the reservoir-rearing fry rely on thermal stratification. They rely on there being a cold layer, and then there's hot water further up. And so we also know a little bit about fry survival from this is a pretty cool study that a friend of mine came up with using genetics it's a cool statistical model. I'm a statistician, so I'll geek out on this a little bit, but we found a way to estimate survival for these really small fish, which typically you can't mark with something like a pit tag. And we saw differences in survival through time and in different years. 2017, that wet, cold year, survival was lower in the early part of the year when fish were smaller, and we think that's because of predation. There's also a chance, I said it's apparent survival because we didn't account for fish going out of the dam, because that could be another source of losing fish. But then in 2018, that hot, dry year, we actually saw the opposite pattern where early in the year survival was higher and then it dropped down despite fish getting bigger. We think that's an evidence of the copepods, which we saw have a lot more of a prevalence in that year. And so we do know something about survival. We also know something about variability in time over years and within years. And then finally, we're going to get to dam passage, the reason that we're sort of talking about these operations we talked about earlier. Under historical operations, most fish in the years that Matt Keever looked at this passed in the fall and the winter when the reservoir was low, when fish were able to get through the turbines and the regulating outlets. I don't know that there was a lot of spill in the years that these were studied, but the other things that we know is that these are not great routes for fish to go out. We had numbers of fish. We also had relatively high mortality in these fish, particularly for larger fish. So we know that the way fish were passing under historical operations was not good, right? So passage was poor. So review, what have we learned? There's 3 different migratory pathways to Lookout Point Reservoir. The spring subyearlings, which we spent a bit more time on. Those are the reservoir-rearing fry. The fall subyearlings, which would probably encounter those same passage conditions, those poor passage conditions that are historic operations, but they would be smaller fish. We also know that reservoir-rearing fry face greater threats of predation of disease, but also have greater opportunities for growth. The relative risks of these things depend on environmental conditions and dam operations, and those will change from year to year, okay? And we know that fish passage under historical operations was poor. So that brings us to the injunction and the management, the new management operations that Greg talked about. And I also want to thank Greg for bringing up the rule curve, because it means I don't have to spend a lot of time on here. But this is for Lookout Point rule curve. And there's 2 parts to this really. The rule curve has changed in 2 ways. One is there's this spring spill period because we want there to be a surface spillway route. And because of the way the spill gates open at Lookout Point, where they sort of open from the bottom up, if that makes sense, the idea is to lower it down so that they don't have to dive down to find where the water's actually going out. So that's the spring spill curve. And then there's the fall deep drawdown. And because of various constraints, because they couldn't drop it down too fast, that started relatively early with the goal of getting it low enough later in the year, right? So this starts in what, is that June or July? But it's a long process to get it down. And here's what that looks like from space. So this is June 1st of this year. That's near the peak elevation for this year. That's near those surface spill levels. It's a false color picture, but you can really see where the water is and where it's not. This is not as high as it would be in a normal wet year. And then on the right, that's from just a couple of weeks ago there. You can see that turbidity that Greg was talking about. You can see how much less of an extent there is to the reservoir. So that's pretty close to that 750 feet. That's about 16,000 kilacre feet target. So this is what it looks like. So we are actually doing a telemetry study right now in Lookout Point, trying to study fish passage and particularly survival underneath the drawdown. I can't say anything else about that because the fish are still in the water. We just don't know yet, but stay tuned. That's coming up. And again, this is kind of harking back to what Greg said. There is some early data that suggests that these operations have changed what would have been the typical environment that the reservoir-rearing fry faced. There's some early evidence that the reservoir went isothermal, that stratification broke down in August. And so it was hot throughout. There was no cold water for these fish. It's preliminary data, so don't quote me 100% on that, but that's what we're seeing. And so you can kind of see this was actually a pretty cold year. There was a lot of snowpack, particularly earlier, April, May was close to the highest on record. It was kind of a hotter early part of the spring though, but what you're seeing is there on the bottom plot on the left, water temperatures in June, July were below the dam, were some of the coldest among that period of record. And then by August to September, we're getting to some really hot temperatures. So these operations, we saw a different temperature profile this year than we saw in previous years. Granted, every year is different. Granted, I can't separate out necessarily what part of this is environmental and what part of this is dam operations. But there's evidence that these injunction measures have changed what we knew previously that these fishes experienced. And so that brings me to what don't we know? So actions to improve fish passage, we need to consider those in the context of these different life history pathways, with the understanding that we don't actually know which of these life history pathways is dominant, if any. Possible effects of the injunction operations, safer passage for for fish in the spring, both yearlings and fry, maybe safer passage for fall sub-yearlings, either those fish that enter in the fall or those that hung out and survived through the fall. Thank you, Yvonne. There's some evidence that there was loss of stratification. Again, not 100%. We know that there was increased turbidity. We don't know how that's going to affect fish survival and within the reservoir and past the reservoir. We really don't know the impacts on the rearing fry because we don't know how many of them passed in the spring spill injunction if they did, and then we don't know how many of them survived. We don't know any changes to dexter temperatures and the fish community there, and we don't know the longer term consequences for reservoir ecology. Chrissy talked about that a little bit this morning, but it could be that this changes the community the next year. Maybe there are fewer predators next year. This is not a one and done thing. So there are potential long-term consequences to this operation. But what I do want to emphasize is that if we're going to assess the effects of these operations, we need to ensure that we're studying in a way that's representative of the natural origin fish. And that's difficult to do for these fry because they're kind of too small to tag with some of the technologies we normally use. Our telemetry study is probably more representative of those fall sub-yearlings that come in because we release them above the reservoir sort of September, October. So we'll get a sense of how the operations affected those fish. We don't necessarily know how it affected the fry sub-yearlings that enter in the spring because we don't know if they survived to pass in the fall. So these are things we need to figure out as we move forward with these operations. And we do need to consider interannual variability. So I pointed out this was kind of a cold, wet year, a little bit drier though, total precipitation was a little bit lower. And then there was a hot part in the spring and a hot part in the fall. Different years are going to produce different things. And, we have a fixed rule curve. Is that the best in every year? We don't necessarily know that. And so we need to consider interannual variability, and we also need to consider something called the portfolio effect, which again, I want to thank Chris because he brought that up and he made me put in this next slide, because there are these different life history pathways. These fish have different ways of getting out, and there's a reason for that. It's sort of spreading the risk. One year might be, it might have been good to be a fry migrant. Another year, it might have been better to hold over in the spring, right? That's how these fish sustain through time, through interannual variability, because they have this portfolio of phenotypes, and one might be successful, more or less successful. We don't know much about the relative contributions within a year, let alone across years for spring Chinook. But I do just want to share one slide from a population that I also work on where we do see this. So this is some of our research on Snake River fall Chinook. So this is natural origin fall Chinook in the Snake River. And what we're looking at here is basically smolt to adult returns through time over about 30 years. And what we see in this population, which is kind of a nice analog for the Willamette Valley Spring Chinook, is Snake River fall Chinook also have a couple of life history pathways. They can either be sub-yearling migrants. They can rear within the main stem Columbia and the reservoirs and go out as yearlings the following year. And what we saw when studying this population over time is that in different years, one of those life histories was better. They had higher smolt to adult returns. That's in that middle panel there. You can see sort of in the 90s, the green is the yearling life history pathway. That actually sustained this population. I'm sorry, the purple is yearling. And then later on, it was those sub-yearlings that became the much more successful population. So this is that portfolio effect in action. And I think we need to consider that with the Willamette Valley Spring Chinook. We can't just prioritize one of these pathways, even based off of 1 year of study. So that, I just want to acknowledge some folks who are, especially the folks at the bottom there, who helped us with the telemetry study that went on this fall. I want to thank folks at Oregon State who helped us put that together. This project was funded by the U.S. Army Corps of Engineers, and so I want to thank them as well. And with that, I will take any questions.
We have some time for quick questions. Can I use a microphone?
You touched on temperature briefly, and maybe other people would have information on this, but has there been any modeling done on the temperature impacts of the reservoirs itself? You know, if you reduce the surface area of the reservoir, does that decrease the temperature enough to offset some of these spikes, for example?
Laurel is one of my co-authors here. She might be able to speak this a little bit better, but actually my understanding is that less volume is actually going to result in warmer temperatures. But Laurel, please correct me. I'm a statistician. She's the hydrologist. You got the microphone.
So to answer your specific question, we have modeling. We've modeled the reservoirs quite extensively. We haven't. Oh, that's better. Sorry. To answer your question specifically, we have modeled the reservoirs to look at temperature impacts extensively, but we haven't addressed exactly what you're asking. We could. In some cases, for example, at Cougar, if you drop down low enough, there seems to be a threshold point where the overall temperature comes down because the residence time is lower. But in other reservoirs, and this is very preliminary, for example, at Green Peter or Lookout, what you are seeing is that the reservoir thermal structure is changing, but it's not necessarily a net increase or decrease. It's more of a timing issue. So you have to trade off thermal conditions would be the short answer. Sort of long answer, actually, sorry.
All right, thank you. Thank you very much. We have time on the during the coffee break. And we have one last presentation for this session about reservoir management with Kurt Carpenter. I think he is online, right? Zoom, okay. Let me see. Kurt, are you online?
Can you hear me?
Yeah.
Microphone is crackling.
Kurt, do you have your presentation? I think I may need to move it to the main desk.
Looks like you stopped sharing.
Yeah, I can see the static. Can hear it.
Can you stop sharing your screen? Can you hear me?
Oh, there you go. I can see you, but I cannot hear you.
You can't hear me?
Yeah, there's no audio.
Okay, let's go to the chat. You still can't hear me? Let's try this.
We try it again.
How's that? Oh, loud and clear, someone said.
Yeah, Chrissy was recommending maybe you can call in.
Okay.
So I apologize from the room. We're trying to connect to microphone is not working very well from his side. So
All right, 1 second.
if you don't have the number, Kurt, the number is in the chat room now. Someone put the number. We will have a student mentorship opportunity after this talk and the coffee. So if you are a student, maybe you can raise your hands just to make sure how many students do we have in the room. Okay, there's one down there so people can see and approach, talk about mentorship. Thank you.
Okay. Looks like we got it.
Okay. Can you try it now, Kurt?
Yep.
No
So
feedback, but less static and
Okay.
kind of
We can hear you now.
low volume. Yeah.
great.
And okay, can you share your screen? I can just put it in the big screen here. I think we can. Can you hear me, Kurt?
Yeah.
Okay. I don't see your presentation in the box folder, so I assume you will share your screen, right? All right. Okay, give me a second, I will share this. All right, we can see your screen, and can you test your audio, please? We cannot hear you, Kurt, yet. Can you check your phone? Because we can see the screen now, but no, don't.
Okay, how's that? I put you on speaker.
You may need to mute
Oh,
your.
that doesn't sound good. How's this?
Yes.
What fun. Okay.
Good. We're good.
Thank you. Can I start?
Yeah, you can start.
Thank you so much. So thanks for hanging in there here. I'm gonna need to mute. Okay. So thanks, Dalton, for sort of setting up our
our season this last year. I'll talk a little bit about that, but I can blaze through that because we've already seen some of that. But today, I'm here to talk about the algae bloom down in the Ross Island Lagoon in Lower Willamette that we had this last summer. I want to recognize a pretty large group of different people that have been working on this issue for a number of years now. And for those of you that have been following, we on the lower right here, we see a color map here of the Ross Island Lagoon, and then in red, you see the bloom going down into the Lower Willamette River. And this has been going on for quite some time with increasing attention from the different management agencies and also monitoring. So I'm going to talk a bit about the bloom from last year, and I'll start it off, and then Nathan Reach from Oregon DEQ will pick it up halfway through. So started here. That doesn't sound good. Can you hear me okay?
Yes,
Can you hear me?
Yes, we can. Can you put in presentation mode?
Okay. Unfortunately, I think I'm going to need to turn off my speaker on the laptop and just run it through the phone. So, okay. So during this talk, I'm gonna present some data from the lagoon here, and you can see my cursor hopefully from the lagoon, and then down in the Holgate Channel here, and then with the stars here, we have continuous water quality monitors. So the downstream one at Marston Bridge has been in place for some time now. It's at a fixed location in kind of the middle of the water column. And then new this last year, we put a monitor here at the surface at the OMSI dock, if you're familiar with the OMSI there on the right bank. And it's parked at the surface specifically to be able to catch this bloom. And then later, and here's a blow up of the map here. You'll hear about some cyanotoxin data that were collected by Oregon DEQ at these sites moving downriver. And as Dalton talked about, this last year we had a robust snowpack. Really, it was a late spring snowstorms that really pushed the snow water equivalents up. And then you can sort of see this from different sites from the Willamette, but sort of Mount Hood and then Clackamas, Santiam, and then down in McKenzie. But that had a big bearing, of course, on conditions, and Dalton showed this as well. But here's a graph that shows our temperature data that points to here at A. You see record low temperatures going back 15 years or so, and then we saw temperature starting to warm up, and then the big drop in the temperature at B when the snow melt sort of peaked. But then after that, it was really game on in terms of growing algae. But fundamental condition, it wasn't a great year to grow algae. So with that context, we can look here at the total amount of chlorophyll at the continuous monitor, and you see here 2023. It was not especially high. We had some chlorophyll levels here in the spring that came up, but not real high compared to previous years. So that sets us up for a spring diatom bloom, where you get a lot of these types of organisms that are very helpful for food webs, green algae and diatoms, and not really the harmful algal bloom or the HAB forming types of plankton that we often see. They didn't really show up until July, really sort of middle of July. Up to that point in time, it was mostly these non-toxin producing oxygen generating species, edible algae. And if they didn't push the pH up to levels that barely exceed the state water quality standard, I would call them a beneficial algal bloom, just to contrast with the conditions that set up during harmful algal blooms. So in the springtime, we had a river turns gold type of situation. You took a plankton net out there and collected a sample, you would see a lot of gold that were the diatoms that were growing in the Ross Island Lagoon, and then coming out into the Willamette River kind of slowly over time. But during some periods, there was a time towards the end of June when you can see here green and blue spikes kind of over the background here
or baseline, where some slugs of bloom came out of the river or came out of the lagoon and went out into the Holgate Channel and then on downstream. And this sort of foreshadowed what was going to happen, I think, 6 weeks later, after we had really an increase in the temperatures and a considerable heat wave. But before that happened, we started to see periods when, here's a better view of that, where chlorophyll levels snuck out of the lagoon and went on down past our monitor. We also saw some cyanobacteria pigments in the form of phycocyanin come out at the same time, but that was mostly due to this sort of stuff. There's a lot of it. It's benthic cyanobacteria. These are mats of formidium, and here's what it looks like under the microscope. And they're toxin producers. Have documented that throughout the Willamette Basin. But initially, those signals really came from the benthic cyanobacteria. So as I mentioned, mid-July, first sign of this here, which is microcystis, and it's a toxin-producing cyanobacteria. We started to see that in the plankton net toes in mid-July. And then Oregon DEQ shortly thereafter got a complaint that was issued or sent to Metro that came around to DEQ, and that prompted some toxin sampling that Nathan's going to talk about. Here's some 20 micrograms of microcystins, which is above the criterion. A little bit later, early August, the operators at Ross Island Sand and Gravel started to notice green scum and sent in some pictures. And so we were, very much on the lookout for when this bloom was going to be, making its way out into the river. And so here, this graph on the lower right here shows the total chlorophyll on the first y-axis here, and then phycocyanin in blue. And I really want to key in on that. So here was this early spring period with the diatoms, but then about a week before the bloom entered the Holgate Channel, we started to see the phycocyanin levels starting to spike. And then this one big giant spike here was a period when the microcystis came out of the lagoon. And here's a graph that really shows this quite nicely here. So what you have is the discharge with the black time series here, and this is at the Willamette River, Portland, just downstream at the Morrison Bridge. And then in blue, we see the phycocyanin levels, and this is just one day. So it happened to be here on this August 15th date where, and in black, you see the tide. So, and this goes from positive, 30,000 CFS down to negative. So, really strong tidal effect in the Willamette. And so if you just sort of follow the day, the tide coming in, and then the tide starts to go out, becomes positive here. And then right here at the end of the outgoing tide, kind of like right in part of this time series here, we started to see these three waves of phycocyanin coming out of the lagoon and down into the Holgate Channel and on downstream. And then the flow, once it drops below here, it becomes negative. And so that starts coming back upstream. And so we see the echo of these spikes going back past the monitor here, much diminished. So it comes out at the surface, and then it probably mixes down. And then when it flows back upstream past the monitor again, the levels are much lower and diluted because of that mixing. And then this graph shows basically the same thing, but then also now with the chlorophyll in green. So after the bloom, which again has both phycocyanin and chlorophyll, of course, when that starts to come back upstream, you see the green peaks are actually higher here than they were here. So this tells me that the green algae, diatom, cyanobacteria transport out of the lagoon, go downstream past the monitor, cyanobacteria get dispersed and mix down, and then but the green algae and diatoms continue to grow and produce these echoes that are offset in time, but as the bloom flowed back upstream. So that's all very interesting, and we've been sort of tracking these spikes in chlorophyll and phycocyanin down at Morrison Bridge for some time now, and wondering if they were real or what was exactly happening there. But and having data from both of these monitors, it's really showed us the dynamics of how the tidal effects influences these populations. And these spikes are real. These little red circles show periods where we see increases in the pigments, and you can see that they correspond to the downward spikes of the black lines here, which is the discharge. So the bloom passes the monitoring station and then continues to grow as it goes downstream, but then it stops and then flows back upstream. So it's really a quite dynamic situation, but that can help us understand where the bloom might go, how it might progress, and those sorts of things. At the time, we also saw some considerable effects on water quality. I mentioned earlier with the pH with the spring diatom bloom. Over here, we see kind of in this orange time series here where it sneaks above the dash line, which is the state standard for pH here. But during the peak of the bloom, pH levels were up to almost 9.6. So that's screaming high for pH. You get above 9, and some studies have shown damage to fish gills. This is a point that I brought up earlier in the talk about the parasitic copepods. But definitely seeing impacts on pH. The dissolved oxygen levels getting up to 176% saturation. So just really cooking. And during that particular time, that heat wave, temperatures at the surface of the Willamette were up over 27 degrees. So certainly seeing considerable impacts on water quality from these blooms. So if Nathan is there, Nathan, you're there to join us?
Yeah. Hi, Nathan from Oregon DEQ. Wanted to start with a time lapse. This is using USGS's REACT tool. It uses Sentinel-2 data at a 10-meter resolution. This is during the time period of end of July and end of August. And you can see the red is showing that flow coming out of Ross Island Lagoon. This is a Sentinel Hub script that produces a heat map for chlorophyll A from microresistant species. So while not as dramatic as the REACT tool, it gives a more precise for our uses. And you can see that same time period, that same bloom leaving the reservoir. Next slide. Kind of zoomed in a little. This is the high point of the bloom within Ross Island. You can see a lot of that activity is around the shores of Ross Island Lagoon. It's a pretty deep lagoon, so it's kind of taking up the nutrients along the shoreline. I want to really quickly get into a brief summary of what DEQ does for recreational HABs monitoring. It's a program that's in support of OHA's program. We mostly do the sampling and analysis. OHA uses that data to decide on any advisories. And it's based off of kind of an intersection between recreational usage and the bloom severity. So that's what we look for when we're looking to take a sample. Where are people most active and the bloom is most active, is that's where the threat to human health is the highest. It's a completely reactive, passive monitoring program. So if a complaint comes in or we see something on the satellite, we go out and